Dear Vice Rector Brigitte Felderer, dear speakers, dear colleagues, dear students, not as many as I can see, it might be still a bit too early, dear audience, welcome back to our second day of our symposium, Cultivating Matter, is the Future Alive, where we explore how architecture, design and research can contribute to a more sustainable and regenerative future. Before I'm going to introduce our keynote lecture, I would like to go through the day. We start off with Joachim, which I'm very happy about. Then we're going to have 3 sessions. The first one is about prototyping and fabrication, which is led by Sándor, who's sitting here in the front row. The second one is about bioreceptivity, run by Barbara Imhof over here. And the third one is critical reflection by Gonzalo Vaíllo, who is, dear Gonzalo, over there, that you can see. We're going to have closing words by Camille Breuil, who was intensely involved to do this symposium and make everything run smoothly. And at the end, we are going to open the root exhibition and presentation, and we'll be closing with a small reception and an aperitif. It is my great pleasure to introduce you today to our keynote speaker, Joachim. He's an architect, urban designer, and one of the most forward-thinking voices in ecological design. He's the co-founder of Terraform One, a New York-based think tank and non-profit design group dedication to advancing socio-ecological urban futures. Terraform One has become widely known for its radical, research-driven approach that blends architecture, biology, and urbanism to imagine cities that not only minimize harm, but actively support diversity and planetary health. Joachim works challenges conventional boundaries of our disciplines. Whether exploring mycelium-based construction, speculative mobility systems, or habitats designed to coexist with other species. His project asks us to rethink the role of architecture in an era of climate crisis and mass extinction. His work has been exhibited internationally from the MoMA to the Venice Architecture Biennale, and he is recognized for pioneering approaches that bring together science, activism, and design. Today's keynote, Design Against Extinction, speaks directly to the core theme of this symposium. It invites us to consider how architecture can move beyond sustainability as damage control and instead become a catalyst for ecological resilience. Please join me in welcoming Mr. Joachim.
Okay, hey, good morning. Hi, I'm up. It's great to be here. Love Vienna. Thank you for the invitation. Do this thing? Yeah. I'm going to share some of my work and tell you guys what we're up to in Brooklyn, New York. So we're a 501c3 national nonprofit, and we've been around now going on to our 3rd decade. This is our book where you can see more details on all the projects we're talking about today. There's a lot of information in there. If you want a free copy, which the publisher hates, and we're a nonprofit, I will happily send you a free copy. Just send me an email. You can find that on the internet. This is the problem we're facing. I guess we're all looking at this. The United Nations thinks by the end of the century, 11 billion people on the planet. How will they move? What kind of waste will they produce? How will our cities thrive? And the issue we'd like to deal with as a nonprofit is the billion other species we share this planet with, which is all the wildlife that's there. So 50%, more or less, of every bird, fish, giraffe, you name it, creature on this planet has more or less disappeared since the 70s. Most wildlife on the planet, 95% or over, is humans and their domesticated animals. So it's not wildlife, that's the opposite, that's just animals. And about every 14 minutes or so, we lose another creature forever. That's a big problem. So we work together in this kind of team that looks at the project of the city, geographers and musicians, architects, all of us arguing and imagining together what our future city might be like. What's the best possible scenario? And it's great to have diverse opinions and to come at it from the opposite view of elite architects and planners. And we do things like this, which is kind of reimagining our city streets to meet those same demands of folks that are there coming to the table. So this is the idea of 42nd Street, where it's as green as possible. And I'm going to get into this in a little bit, but we have this new notion of the value of that green. And that's going to radically change, and we're going to have a personal connection to it. But here, it's for air quality. It's for food production. There's a riparian corridor going through the center, teeming with aqueous life. We have trackless trains, vertical access wind turbines. It's a version of the city that we think we should get to, as opposed to the previous century. And some of our biggest influences. So I thought, it's a university. I'm going to talk about the reasons why we got into this work. So one of my favorite Americans, Henry David Thoreau, I would say was the first major intellectual to not come out of Europe at the time. And here he's thinking about, let's flip the script. Let's change the narrative to not be about the drama of the human will, but to be about the kind of the activities that happen in nature. The kind of the quotidian in nature becomes the centerpiece of our novel. So the reflection of leaves on a pond, a gossamer strand of a spider's web, this becomes the central figure. And to live life deliberately is another Thoreauvian concept. And then a little bit further, more influences. Here is a president of the United States that I actually adore, Roosevelt. And he took time off running the country to go to Yosemite with this man, John Muir. And he was there for about 5-6 days, camping in the winter and looking at some of the most beautiful landscapes one could find. And he created, essentially, the preservation movement, which is different than conservation. Conservation says we could take some trees out, use them, and then plant 3 or 4 trees back. That's okay, but preservation says we're actually not smart enough to do that. Actually, have to leave this land alone. Let it be fallow. It's the land of God, and maybe in the future we can understand it more comprehensively, but for now, let it be. And then Strauss and this endless growth fallacy, which is all of capitalism. There seems to be no limit on how rich you could be. Now that we have Elon Musk will be a trillionaire. And Jane Goodall, another big hero of mine, telling us that other species, in this case chimpanzees, are sentient. They have families, they have emotions, they make tools. We share the planet with them. We need to care more about other species. A brilliant influence. Then this one, which is a little bit hard to explain, but is the reality that we're in. Why do we still use fossil fuels? I know in Europe you're not as bought and sold as we are in the United States, but there are companies out there that will say publicly, I love your concept of this new kind of fuel. It's great. It produces no carbon. The effluent is water. It's a great concept. We'll even support it financially. But they won't let it scale. Predatory delay is a public face that says we support some concept that will benefit the environment and help to work positively towards climate change, but behind the scenes, they slow it down. Because every single day that they're in business, the profit is enormous. It's a reality that we're facing. And then Timothy Morton, for those students who haven't read some of his books, quite an activist, philosopher, big thinker. He has this concept called OOO. That's how you say it. It stands for Object-Oriented Ontology. The chairs that you're sitting in aren't just chairs. There is an entire ecosystem, entire life behind them of people and communities and resources that have been extracted and used to make that chair. And the idea we don't treat it with any level of interest or equity or equality is part of who we are, but we need to think deeper about where our products come from and the resources that are used in the communities that are behind them. This is also goes back to Arnes, a Norwegian philosopher, which is where he takes a lot of work from. And then the CEO of Greenpeace, Annie Leonard, absolutely adore her. Here she's talking about this stuff. Why is my, my phone no longer good? we design things to eventually, or we engineer things to eventually need to be replaced when we don't have to. Now, the battery in this one, you could buy the battery, but still, you know, it fails, and maybe they're getting better, but the software in there is about keep on purchasing, buy something new, update it. So if it's not engineered to fail, it will also be perceived obsolescence. It's no longer the coolest iPhone. I don't care about that, but others do, and they'll purchase things to make up for it. So this all begins to develop this kind of single overriding predicate, this theory about design against extinction. And what do we do with these new technologies in biotech that are coming on board as architects and urban designers? How do we use them in a kind of logic and strategy to work well with this planet and to be citizens responsible for the things that we're doing? This here is one of the last northern white rhinos. It had been sort of restored, this fetus, in the San Diego Zoo as they used in vitro fertilization and a surrogate mother to produce this young baby rhino. It didn't live, but science is working to bring that species back from what's called effective extinction. And then this field, or this kind of study, becomes multi-species design. Not just designing for anthropocentric concerns, but for other organisms as well, equal to us, perhaps, and makes it very difficult to get clients in some cases, and I'll talk about that in a second. So we work with all kinds of experimental materials, and I love seeing what's going on outside and all the talks from yesterday. I know I'm in the right place when there's a lot of discussion about mycelium. We have been working with mycelium and different types of biocomposites now going on 20 years. We print them, we use crochet, which is the opposite. It's a very analog product. It's not robotic or computational at all. We use different types of bioresins, cast all kinds of materials, make a lot of mistakes and failures, but eventually try and find things that stick. And then test the organisms, these microhabitats, and see if they like them or not. And in many cases, we're just simply not sure. Or we'll call up a world's preeminent entomologist and say, what can you tell us about butterfly habitats? They'll say, we've never talked to an architect about that before. And we're not exactly sure. So here are the indicators, work towards those indicators. So this is a project we did. It's called the Anti-Extinction Archive. It takes a species that's at the edge of life, a red-listed species. We put in fertilized eggs in a cryogenically preserved chamber that's inside the egg. The egg is designed to slowly decay over 7 years. It's surrounded by the necessary biomass to keep that creature thriving. And over time, it sort of melts back into its environment. People come and visit this egg, and they learn about, oh, this is something that's disappearing. Is there something I can do? Yes, when you vote next time, vote for the right person in office, and perhaps we'll save that species. And then the creature is released. Certainly, this is not something that would save that individual organism from disappearing, but it does build awareness. We got to do more than just do that. We also look at the metabolism of cities. If I was a New Yorker, I would think that New York is designed, sorry, if I was an alien, I'm not an alien, I am a New Yorker. If I was an alien looking down at New York, I would think it's designed to produce waste. 36,000 tons of trash per day. What is 36,000 tons of trash? In about 1 hour, it's the Statue of Liberty of compacted and inorganic and organic waste. That's an enormous amount of product that we make. So we look at not only explaining that and building awareness, but then producing local shows. Here's with the new museum in New York. This is 15 minutes of waste produced in styrofoam down in the East Village. We'll produce these maquettes. And then looking at, is there alternatives to this waste material, the stuff that we throw out? So again, for the students, this is the entire history, and we're near the Mac, the entire history of chair design in one slide. And I think it's pretty useful. So there's the 19th century and before, which is bespoke, handcrafted, custom-made objects where there's a relationship between the maker and the thing that you make. And then there's a 20th century, the Bauhaus, Charles and Ray Eames, industrializations, different types of steels and plastics and modularity, absolutely beautiful, exquisite designs. The reflection of that is seen in the performance of the materials. And I love that age of furniture. But I think the 21st century is about growing our furniture. It's about producing these things in a lab, things that return back into the earth and are fitted into our planetary metabolism. So years ago, we worked with Acetobacter, or bacteria-expressed cellulose, combined that with mycelium, which was hydrophobic at the time, and got this material that was a kind of a chair. This is one of my daughters, 3 of them, in kindergarten with a chair that Daddy made that you can eat. It's 21 separate pieces of molds, and she's sitting in the chair. It was very complicated to do back then, and since then we've gone on to triply curved structures. We saw some great presentations yesterday about controlling the growth of mycelium into precise geometries. Here it's combined with plyboard, and the agricultural waste here is cotton husk, which is great, acculturated with reishi, which made the surface, and it becomes a chaise lounge, so not just a chair for kids, but a chair for adults. When you're finished with this chair, it goes into a garden and feeds thousands of other forms of life as it composts back into the earth. Then we had this project, which is a client of ours. It's essentially a larvae of a beetle, darkling beetle. You may have seen projects like this now in schools everywhere. But we looked at mealworms. Mealworms, because they eat styrofoam, the stuff that we throw away. They can eat about a centimeter cubed in about 2 weeks. Incredibly slow, but very efficient. They process it through their bodies, their enzymes digest it, turns into a frass, they then eat it again, and it basically becomes garden mulch. So we created a farming system and a series of units for mealworms as our clients to thrive. And then we produced a community sculpture through the Bloomberg Foundation Fund, which produced this in Camden, New Jersey. It's a community with a lot of urban junk space, a very indigent and poorly entrenched community, and they have waste everywhere in their city. Here we encourage people to dump their styrofoam in the biodigester and watch the mealworms dissolve it. Not in real time. I mean, honestly, they're very slow, but the rest of the farm is for producing more of the mealworms. So the whole back is for mealworms, the body is for digesting your waste, and then the top is a clock, a mycelial clock that shows how long it takes for different substances and their chemicals to naturally return to the earth. So the length of time reflects how bad those materials are. Styrofoam could take 500 years or more to naturally be processed back into the earth. So mealworms dissolving it in a few weeks, a lot quicker. And now to kind of some of the new work that we've been doing and part of the things that we've been involved in at the Venice Biennale. Landscape is data. What does that mean? Well, we're in the middle of it, so you could probably help me in the Q&A section. But we're thinking that if you have the capacity to embed in the chromosome and the nucleotides of any botanical information, and you could store an incredible amount of information, how does that change our relationship to gardens, to agroforestry, and to the plant life in our cities? Do they store our memories? Could they store our memories? And yes, they can. Here is the future of iPhone storage. And in a picture of, I don't know whose apartment that is. I was looking for one with a cat, but it's fine. You could store somewhere, well, I'll tell you in a minute, an enormous amount of your data that you keep here on your phone, pictures from 2010 of your kids or a trip you went to in, I don't know, Paris, that you don't look at. Something like 2% of the carbon loading in the world is for cold storage of data, not active, cold storage of data that we don't use. And guess what? You can put it inside plants. So this is a proposition of using both AI and then transgenic modification to store our memories inside botanical life. And this was first done, not with architecture, we are the first to do that with architecture, but first done by an artist named Joe Davis working in George Church's lab at the Wyss Institute at Harvard and MIT. The project was called Malus Ecclesia. Malus means apple, the bad apple. Ecclesia means church, after George Church. Joe Davis, as an artist, took in the history of the Garden of Eden out of Wikipedia, and he stored that in an apple seed. He's one of the first to ever do that. Later on, George Church took his entire book, Consilience, and stored it inside an apple seed. And this is how you do it. you translate zeros and ones into ASCII, ASCII into AGTC, into the base chromosome of a plant, in this case, an apple. You store that in the apple seed. It takes hold and grows into a full apple. All of your information is permanently contained. It does not change the apple. Just because you put Adam and Eve in the apple doesn't know the story, doesn't read the Bible. And it's not going to read the Bible back. You can bite into the apple, you won't feel Jesus. It's just there. And it's replicatable for 1,000 years. It stays in the base chromosome. It's just noise to the apple. Could be noise to a lot of people. You store all of that information. That's 215 petabytes of data per gram of botanical DNA. That is immense. It's three-dimensional capacity of storage. What if you put in all kinds of architectural information and you have a kind of an genetic agroforestry system where you store archives, everything from Vitruvius to Norman Foster, plans, sections, drawing inside botanicals. One of our thoughts was, this is an early one, is you could store that in corn. You could store every George Lucas film, all the Star Wars films you wanted to. This is the amount of 215 petabytes, by the way, is 13 years of continuous play of high density film. Straight. And that's just in 1 gram of plant DNA. So you could take every Star Wars film, all the plans, sections, and drawings of all the spaceships and the Death Star, store that in corn, then grow the corn, make popcorn, watch Star Wars, eat the popcorn, knowing that you're eating the entire history of the Star Wars film, whatever, and then it's stored in, it's not doing anything, it's just in your body. But you could then take that individual kernel of corn, pull out the information, and then start building 3D models of that Death Star. It's pretty interesting. And so we started taking books and genetically attempting to break down books and store the information directly. Came up these sort of memes to kind of describe what we're doing with plants as active places to keep our data without harming them at all, but now having a new relationship and appreciation for this plant life. And these are the facts behind it. That's 330 megatons of carbon a year from our cold data storage, which is enormous. And then for the Venice Biennale, we did a first. We took form follows function, the words, from Louis Sullivan. We converted them into AGTC, put them in the base chromosome of kelp, ocean kelp, and stored inside there, and then start producing these 3D prints of a kelp-based material along with clay and alginate. But stored inside it was all this architectural data that was inside these prints, including the geometry of the things that it's printing, so that you can then take out that information of the print and reprint it because it's stored inside there perpetually. So this is a 3D printed clay kelp combination. Talk about why we're doing that in a minute, but I'll talk about it now. Our sponsors, one of our sponsors, because a lot of this work needs that sponsorship, is Crème de la Mer, Estée Lauder, and their main component that they use in their skincare products is kelp. So that was our client, the kelp was, and then Estée Lauder supports us for 3 years, which is great. And then imagining the use of kelp in streets, using kelp systems that are also transgenically modified to help restore scorched earth, which is an amazing capacity of kelp or any microalgae as it restores burned landscapes. So here we are working on this particular project, creating a mosaic landscape of printed geometry designed to help restore a specific area. This on your left is the AGTC, it's a model, structural model of the chromosome arrangement of form follows function. It happens to look like that when you do the scan, which is great. Then we produced a synthetic reef for underwater usage to store the kelp. And the concept here was to take kelp as a carbon sink, which it is. It's probably the largest carbon sink in the oceans, which is great, and combine it with a data sink. So we have the largest data sink combined with carbon sink together in these synthetic reef systems. So here's the earlier versions of storing information into coral life, then our production of synthetic coral combined with the kelp. You can see here we produced a model that, was it like 6 meters? never get meters right, but more like 18 feet in height. Is a massive system produced for the Venice Biennale with the kelp plants and their genetically modified data stored in the vitrines, and then the rest of it's designed for all kinds of aqueous life and other plant life to thrive around the system and to encourage diversity underwater. And this is it on display in Venice, and now going to the MIT Museum in Cambridge. And just explaining to folks what's happening with DNA just wouldn't work if we had a drawing and a few test tubes. It just doesn't capture the imagination. So we did adjust this kind of extreme points of scale between genetic level nucleotides and then the scale of a massive ocean reef to show the variability and also to express that we're storing so much information, so much data, and also attempting to deal with carbon. And then just a couple more projects, because I only have so much time. So we're working on this one for 17 years. There's some folks out there doing amazing work, and some in Stuttgart. This is about grafting inosculate matter to form one contiguous vascular system, or making diagrid structures with plant life. This plant life also now can be genetically modified, but this is looking at grafting systems or pleaching and growing them into geometries, usable geometries for structure. Not easy to do. Why would you want to do that? Well, if you compare something like a concrete column that's been reinforced, steel and concrete, or a 2x4 that you would purchase from, I don't know what you call it here, Bauhaus or Obi. We have Home Depot, which is like a nightmare in construction in the United States. But that 2x4 of wood that you get, it's okay. It does sequester some carbon. It'll be used, last maybe 80 years, but then it goes back into the environment, rots, and that carbon returns into the atmosphere. When you produce a structure made out of living material, it is a positive contribution model. Not only is it sequestering carbon, but it's producing oxygen for its entire life. And you can have tree systems or botanical systems that could last easily 100 to 150 years and keep those structures active. Over the years, we have made tremendous amounts of, I wouldn't say epic fails, but attempts to accelerate the growth of woody plants to make engineered structures. And these are some of those attempts, including these extended articulated surface materials that tease roots, tap roots of plants, so they grow long and wet in dark ultrasonic chambers. And then once we get that big, long, wet root, we bring it out into the sunlight, shape it over parametrically designed scaffolds, hits the soil, it immediately lignifies, get bark production, leaf propagation, starts to get solid and hard, and you could graft it at that point. Just takes too long to do that. A couple of years to get those roots to grow maybe 2 or 3 meters. So we discovered, ironically, it's been around for a long time, biomass farming. In this case, producing willows, shrub willows, white willows, which are 17 feet or longer in height. They're cut down and pelletized for wood-burning stoves, so it's great renewable energy. It's a dicot, not bamboo, which is a monocot, so it's not a grass. And these are these are essentially tree farms, agroforestry. We could use these to then graft them at full scale and assemble structures at full scale on day one, instead of having to work with all these roots and growing them over time, which I still love to do, still trying to work that out, but without genetically modifying them, I can't make it work fast. This is one of the earlier designs. This is the latest. One of the reasons for that is that Americans, not Europeans, but Americans suffer from affluenza. They just want stuff now, like right now. They don't realize trees don't grow now, and they take time. So we're working to meet those both constraints, the reality of nature versus the needs and economies of capitalism in the United States. So this particular model is a final version of our tree structure system. You could find this, it just opened in October in the Museum of Natural History in Bern, Switzerland. It'll be on display for 23 years, along with our chair and some other things that we produce for the museum. But this is showing the essentially, whoops, I can't go back. Oh, I could go back. This is showing the kind of the ultimate fit out of the structure. It is not designed for humans first. This is a multi-species structure designed as a terrestrial reef or a land coral to encourage biodiversity in forests or woodlands adjacent to cities or in the peri-urban conditions in and around cities. It uses scaffolding, in this case a treated CLT, that the plants are woven to fit the geometry. Each one is located parametrically, and then the scaffolding is removed, reused on another structure after about 5 years of growth. It's also a double-tiered connection. So we have a connection from the neck of the trees below the crown to the root structures. So a 17-foot tree plus a 17-foot tree makes a very tall tree on day one. This is showing over time. Here it's 10 years. Actually, we think it's more like 5 to 7, but we remove the scaffolding and all is left is the modules in between. Those modules are microhabitats for food webs for different multi-species to engage in this land or terrestrial reef. So the terrestrial reef is filled with life. On day one, as we did the installation, one of the teammates, she screamed. And I was like, oh, what's our insurance? Like, what happened now? Turns out we had a family of frogs moved in within an hour on site, which was pretty cool. And then I think they were eaten by something else because there were snakes and insects, there are wolves, there are bears, there are all kinds of deer. We now have 2 years of research with multiple wildlife cameras capturing what the intensity that this brings to that area of the forest as wildlife takes over this fecund environment. This is an idea of pre-planting a village of these structures. Eventually, they'd be converted on the interior for human use, but a completely separate structural condition, not exactly, not designed for human programmatic use, not for multi-species use. It's also a diagrid, which is really important. So the branches connect to branches. They cross over here at these X zones, and they're woven all together for lateral loading and bearing, making an incredibly strong, complex woody plant structure over time and get you some idea of scale and how things are growing. So we're constantly pleaching them. We have arborsmiths, which are sort of like, they're tree sculptors, working to kind of gently nudge, not choke, but nudge the branches and tree elements into place and to respect them and to keep their geometry. We looked at these habitats and thought of all kinds of modular design elements to respect everything from avian life to reptilian to insect, et cetera, and larger mammals. Here you could see the diagrid beginning to take place here, and then these are the habitat zones that were made out of crochet. Why crochet? We saw yesterday, you can weave, you can knot, you can, there are a million ways to do all kinds of fabric-like components. Crocheting is still not automated. I think there's one robot in Brooklyn that can crochet. Crocheting is only handmade, and it's crocheted jute with a rosin, a pine rosin, and a beeswax to keep it stable against the weather. All of these are handmade components on a printed scaffold. Some are a bioprinted system, and then they're placed inside the structure there. This one is showing you that eventually the interior will have things like toilets for humans and a shower and a bed area, and then very thick double skin facade made out of hay bale, et cetera, on the inside, which we'll do later. This is it, looking very Blair Witch on day one in like the early winter, and then the plant life starting to take hold and grow. It's, for those of you who are landscape architects, I mean, you do the drawing, and then you have to wait years to eventually get the photograph. It's the same with us. We had to wait some time to get the photos we wanted of plant life and all the mammalian life taking hold. So here it is, a winter day. You could see one of our team members here for scale. It's a very, very large tree structure. And then in the summer, where everything is starting to blossom and bloom in and around the system. And that's the interior. Every arch is different to look at the different pressures and PSI strength of all the different weights of the trees being grafted and assembled together, the form at the top. And then another client of ours, this guy, and thousands of these guys, crickets. Here we were thinking about replacing eating cows, pigs, chicken, and lamb with eating bugs. That was the United Nations, 2013. Why eat bugs? So, anyone know why you want to eat bugs? Is it too early in the morning to think about eating bugs? Because it's around 2,000 gallons less water for the same gram of protein you get from a cow versus an insect, 300 times less greenhouse gas emissions. We wanted them to taste good. So we used a technology called architecture, space, light, air, designed volumes for insects so they'd live these amazing, happy lives. In this case, crickets, 6 weeks of life. They die naturally, and they're harvested, and then we turn them into a flour. That flour makes bagels, bonbons. The best pasta I've ever had came from cricket flour. It was a Michelin-rated chef from Italy who made the pasta, which probably helped, but it still was fantastic. It has to taste good, otherwise people won't work with it. And we have a patent on this system, a dial-lock gate, for as they get bigger and fatter and older, producing lots of kids living inside the system, and they move throughout the shelter, spreading their genetics. These systems on the top are called cricket horns. It accentuates the sound of cricket chirping. When the males are horny because they're not worried about surviving, they move their legs back and forth, stratolization, and they make a sound chirping to find females, and then they reproduce. And here are these cricket sex pods somewhere here. Here they are, where the two of them get together, they do their cricket thing. I never like telling this, it's always so odd. But it's a brothel, I don't know what it is, for crickets. And it works very well, and it's the only architectural use of a Voronoi pattern that I've found successful. And then working on something like this is a pod that produces food locally to go from pasture to plate. So here we are using these all foldable geometries, including the main structure, which folds up into not a green wall, but a green ball, a sphere to grow your food. Spirulina, wheatgrass, arugula, not something like corn or wheat. We've got big farms to do that, but stuff you want to have fresh and local to put on your plate, and that you would grow and have a relationship between the things that you grow and the things that you eat. It's part cabin, part sofa, part farm. You can put it inside your home. It has grow lights, spins on wheels, gets all kinds of solar income, and everything about it becomes flat packed and is shippable. IKEA has one that looks nothing like this, as enormously cheaper than ours. Great. I guess I want to end on like this thought about vertical data gardens and all those renderings we've been seeing for years of the promissory notice of having plants on the surfaces of our buildings, and what exactly are they doing? Some cases air quality, maybe it's for food. A hero of ours and a longtime collaborator who just, he passed away this year, Dixon Despommier, sort of the godfather of the vertical farm. Here we have a vertical farm in a city doing its thing with microalgae and all different types of plant life, but it's also for data storage. So it gives us this new sense of, oh, there inside these plants are our memories, our data perhaps of a civic space or some event that was important. It's not just on the surface of buildings, doing a little bit with air quality, may be pretty, but it's actually contributing to the to our memories as an archive. And then I know I'm getting out of time, so I could see this. These are blimps that do the same thing. This is a hard drive in the future. Not going to talk about it. And last thing, saving the monarch butterfly. So these are painted ladies, not monarchs. We were commissioned by the American Museum of Natural History in New York to work on feeder systems, to think about how kids come and relate to butterflies. The monarch butterfly, the New York Times just did a piece on the airplane. I was reading it. It's disappearing in the United States. They are native to New York, and they go to Mexico over generations. We now could track them with a solar micro tracker, which weighs about half a grain of rice. So you stick it on the wing of a butterfly, and you can trace exactly where butterflies are going. So a few scientists are working on that. We've been trying to connect butterflies and humans and vegetation, so there's some sort of reaction, some lighting system, when they are laying eggs or eating or in a chrysalis stage, there is a kind of a focus and a spectacle for humans to be interested in what butterflies are doing. Another project in Los Angeles is about the El Segunda butterfly, here for a tower that's been ongoing now 6, 7 years, but it's another butterfly sanctuary fit into the skin of a building. So we don't lose a single square footage of rentable space, but in the interstitial space of the skin of the building is a vertical meadow teeming with life designed for monarch butterflies to thrive. When BASF was another funder, created this concrete called Green Sense, a fly ash impregnated concrete that's structural with the geometry designed for the life cycle of monarch butterflies, mud bathing, egg laying, meeting other butterflies, interacting with milkweed, their plants. Then a 2-ton engineered facade that was at the Smithsonian on display for a few years to fit into a building that we're doing in Lolita. And when you look out the window of this building, instead of seeing noisy cars and traffic, you'll see that farther away, but there's about a meter of space with this vertical meadow for butterflies doing their thing inside that kind of in-between space. And here's the installation. We didn't keep the monarchs inside there because it's a porous system, so they're able to move in and out freely. You don't want to cage butterflies because it affects their genetics, so they become weak, and then they go into the environment, and it weakens the entire species. But here they are doing their thing. Okay, that's me on time, but out of time. Thank you very much for being here in the morning. Thanks for listening. Happy to answer some questions. Thanks, guys. Thank you.
Thank you very much for a really, really stimulating lecture that you gave us. I'm sure that there are lots of questions. So we start in the back with the first person.
Hi, thank you for the lecture. I wonder if you work in relations or with a specialist with biologists. So because you are producing, or your work produce a lot of, I will not call it artificial, but also not natural, so let's call it hybrid environments, where you attract a lot of animals, insects, and so on. And I always wonder how this hybrid environment in the mid and long term actually affect those animals. So I don't know if you have been tested that or have discussions with biologists, or biologists have to some extent an influence in the design before setting everything up, or how do you handle this thing?
Yeah, love that question, and absolutely. So we have a lot of ex-biologists, people who studied for 4 or 5 years, and they just skip over and for some reason want to do design. Then we have biologists. Our chief science officer was my roommate in school, Dr. Oliver Medvedik. He runs the bioengineering department at Cooper Union. And then Sebastian. We have a team of biologists that are either full-time or there all the time to suggest, to strangle us, to nudge us, to say this is wrong, this is right, or we don't know. Or here's some paper, do a review of the literature, this is the best we can get to. We always look up experts in the field for the specific organism or ideas, and then interview them and ask them, how might you do better? What is the expected outcomes? What is the kind of the quantitative data behind this kind of work? In many cases, it's a great meeting of the minds, but often in biology, there's just not enough intersection with architecture or urban design. There's just those conversations, even now after 20 years, are extremely rare and not often occurring. So we believe in best practices. All the approaches are treating any organism as absolutely sentient and equal, and work towards stopping the insanity of extinction on so many other species. So while if anything produces harm, we immediately get rid of it. And that's never the case. I mean, we're just not that group of people. Sometimes things don't work the way we want, and actually all the time things don't work the way we want. The butterfly research was 7 years of trial and error to get butterflies to drink. And no one was trained in architecture school to do that. And we're still not sure if that works. I mean, they're working in the museum, I guess, doing this kind of stuff. But that's not a hard one. When you put it outside in a field or a forest, you're then releasing this information, and then we have been studying now, going on our 3rd year, of the interaction between different species. And we're worried about predator and prey relationships and food webs, and it seems that one species learns the environment better than others, and that has an advantage, and then you don't get a consistent kind of take. In other words, it becomes a main habitat for snakes, and other things don't survive. But it's an ongoing, nonstop experimentation and research endeavor, but absolutely have biologists and ecologists also, and experts in those particular fields. So working harder to get more grants for those people to be on board in the beginning, but in many cases in science, there's very little reward for doing that kind of research. You're specifically involved in working on your organism. So a great example is caddisflies. There's 2,000 variants of the caddisfly, and they produce these amazing underwater architectural larval cases. There was a famous artist who but only gave the caddisflies gemstones and gold. And they would use that to make this unbelievable structure by themselves, an embodied kind of structure of a larval casing for reproducing, because it's the only thing they had in their environment. When we contacted that person, they just tried it out. When we contacted a leading caddisfly expert in Berkeley, California, and said, what is the kind of the architectural needs for the production of the caddisfly casing, she wrote the book Underwater Architects. Had never talked to architects, just looked at how the casing was shaped from the perspective of a bug scientist. And we had all these discussions of what we could do with caddisflies. We're still ready to do that research, but that's been years in the making, because we're just not sure what to do with it. And I think making them produce jewelry out of gold is not us. I don't think that's exploiting the insect per se, but maybe it is. I don't know. We're very interested in the bioethics of these things too. But does that answer your question? It's a non-stop process, and yes, there's many biologists on the team.
Can I just jump on that with another question?
Yeah.
Also coming from the landscape perspective, so you presented a lot of designs that are very developed and planned, and I was wondering if you also quantify and monitor your prototype over years, just to understand as well the impact on the habitat, as you mentioned, and also to maybe be surprised of what you have planned and how it impacted this species, for example, in the forest with this pavilion.
Yeah.
And how this changed your design?
Yes. We're always monitoring the prototypes, and we're also trying to unpack the data into something that's useful. When it's out in the field, it's not a controlled model. So when a family of bears moves in, they just live underneath it, and we thought that might happen, but there you go. And then that it doesn't disrupt, but affects all of the other work and information and data feeds that we were getting for the structure. And then they go away for some other reason. Plus, when there's a family of bears, you really can't go near the structure to do any more research. I'm not a big, I don't know, I don't go near bears. I'm a New Yorker. There's none in Brooklyn. But so yes, we're often surprised. We capture the data. We do our best to learn. We hope others can find more, I wouldn't say lab-based experiments, but focused areas so that each and every species has its own team of architects, landscape architects and urban designers to figure out how we can work with this creature to give it something amazing. Crickets were a little bit easier. There was a lot of research on that. Anything that's through animal husbandry or things we use like honeybees, enormous amounts of facts and research on that. We don't use honeybees for that specific reason. There's just tons of, we know that one, but when it comes to anything else, no, less and less information. So yeah, we do our best to record and then publish that. All of it's available. I can't say that there's always a net positive effect, it seems, but until we can, just in the New York Times today, we just learned how to count butterflies. And the Monarch, which has entire nonprofits set up who are in love with this species. It takes a while to figure out these systems. And only recently we're able to actually track where they migrate and to have actual data. And that is like, I don't know, 9 or 10 PhD researchers, 2 principal investigators, research funds for years to develop that solar tracker for that specific one. So we're understanding further. I wish I could say that it was our nonstop round-the-clock expertise, but we're pushing forward with the architecture side, little more so than the biology side, I would say, to be fair to the work that we're doing.
Hi.
Hey,
Yes.
butterfly researcher. Yeah.
So, knowing your work, I have a very specific question about the new work that you were showing. The Wikipedia apple, I call it for now, the Adam and Eve
Yeah.
encrypted. So, how does the data stored in the DNA of the apple seed change over time? Is it changing? And if the apple is, let's say, mutating in terms of genetics or something, or I'm not a biologist, but some sort of crossbreeding of apple species or something. How does the data change with the DNA or something?
Yeah, no, love the
Like
question, but it doesn't.
But like long-term change of genetics or something, nothing happening there?
In our own DNA, we carry an enormous amount of noise. In fact, most of our DNA is just random as far as we can tell. So it's extra AGTC, that's in random jumbles. Over millions of years, they occasionally produce a new active trait, another finger or webbed feet.
Exactly.
Yeah, but if we put Mozart in you, like, it doesn't mean you're going to play. It's just extra noise, the way that data translates, and it just stays in there in the system and replicates with you over time in every part of you. If we put it in as you as a fetus, stays with you and then replicates with all of your children and beyond. So it's there is some loss, but when we're talking about 215 petabytes per gram, the loss is just immaterial. I don't know what the loss is equal to the amount of loss we get from our own data servers. I trust Apple is storing my photos from 2009. I don't look at them, so I don't know.
Did you talk to Apple about Apple storage?
Yeah.
Sorry.
That's exactly right. Apple should be thinking about, there is a ton of folks, this is about 10 years old, this idea in tech right now. So maybe 12, depending on where you count. There is an enormous amount of thought that the best thing to do for storage is DNA, because it's going to be around 1,000 years from now. The operating system will still be the same operating system. There won't be a software update or patch or replacement. It's always going to, it's the best place to archive your memories. It's just a matter of it's easy to put in, more or less, and we could do that for anyone. So our cell biologists if you want to do that, we will do that for you, whatever you're interested in. It's a little bit harder to extract, because you have to synthesize the base gene to get that information back. So that's why we're not using it today everywhere. It's not like a USB stick where you can, you can't access it faster. So you just have to put it there and let it be. But it stays, as far as we know, indefinitely. Again, like, I haven't tested it for 1,000 years. That is a supposition that we're looking at. Yeah.
So for the future generations, the decrypting will be interesting.
Decrypting, yeah, you could store your passwords in your house plant. Not joking. That was from the Urban Tech Conference last week. So no one's ever going to find, you can't put your if you have cryptocurrency and it's worth a fortune and you want to store your passwords, where can you put that where some hacker can't find it? You could put it in your plant. You can leave it there until you actually need to then cash in on your coins. Sorry, this is stuff that we're thinking about. It's fun. Great question.
So we have 2 more questions coming up, and I think then we need to wrap up everything.
Yeah, my questions, hello.
Hey.
It's more of, I really, really enjoyed the presentation, because I think it's giving us all a lot of food for thought, literally. And I'm wondering about, like, tagging on to the Apple storage and also the ethics of this. When we're thinking of bringing back the rhino, I feel like there's a sort of nostalgia to before was better. Nature was a certain way, it was preserved. We didn't impose ourselves on it.
Yeah.
But nature is in constant flux as well.
Yeah.
And we haven't really asked it. So I wonder, how do we inform our interventions, like the process, without really imposing our anthropocentric view? And should we form some sort of interspecies communications? Because it's still how we interpret as scientists maybe what the species need, and then we can host that. But I'm wondering, it is a big question, but how do we know, especially when we're speaking about encoding our own culture, our own thoughts into other living beings? Some scientists have also started encoding DNA into 3D printed objects, so the other way around. And I'm wondering what your thoughts are on that. I'm just really curious.
Oh, yeah.
I know, big question.
Great question. I mean, like I said, another book I wrote called Supercells, Building with Biology. The entire book I did with Nina Tandon, a cell biologist, also longtime colleague, 20 years. Did this entire book on growing shoes and fun stuff. And like one of the reviews was about the bioethics of it. And the review was almost longer than the book. It was a very earnest, meaningful review. But it just went down into the morality of experimenting genetically with all the kinds of things that we're doing, and many others. We're not the only ones. And the implications of that. Mostly it was a mixed sort of like decision on that, because it's not final. There is a trust that those operating in this space are benevolent and positive. We are a nonprofit. We're not looking to do this for any sort of reason of greed or abuse. But that doesn't mean that work won't get to that. Or for weaponry design, which sure, so we stay out of that realm. The territories we're working in are explicit intrinsically for support and help and regeneration of ecosystems and ecosystem services. A great theoretician on that is Robin Wall-Kemmermeer. She wrote the book Braiding Sweetgrass, which is a must-read, where she begins to fight with her PhD advisor about the meaning behind how scientists categorically understand plants and other life forms. There, in this case, it's like a taxonomy in Latin names and the specific functionality and the purpose of plants. But then she was saying, what about the meaning of color? Or what indigenous tribes thought of those plants? Or instinctually, what happens in relationships between those plants that humans have known over years and we've lost that information? It's probably a time where we were closer to Avatar and had a deeper connection to the things around us. We've been removed from that. Thank you, Rousseau, the noble savage, this kind of thought structure. So I have an answer for that, which is I know what we're doing is purposely intended to be positive and to absolutely do no harm and to nudge things and do the least amount of work because nature makes it happen. But I also teach entire course on the word nature, because it's a complicated thing. So it is a point of study. I think that the university is here to do this kind of work and to make those points and to argue those points and to feel free to argue those points. So excited about that. I don't have a single answer, except for one of the things we did do is we took the Animal Bill of Rights, in this case for all creatures, and an early project that we had done was to embed the rights of animals into their genetics. So if you go out and you're an American hunter shooting anything you want, inside that, the very genetics of that animal is the legal doctrine that says you cannot and that you've violated them. So it's embedded in this case, it's embodied in their system. That could be very helpful. I don't think it's going to necessarily stop hunters. but it does bring out that dialogue more and more. And you've opened up a Pandora's box of brilliant issues to kind of to tease out and to polemicize. Yeah, I appreciate that.
Thank you very much. It is already 11 o'clock, and in order to be on time, we would actually now start with coffee break, and I would like to ask Andreas to ask the question while having a coffee.
Okay, I'm going. Good morning, everyone. Okay, see you for coffee.
Yes. And
Good. My name is Vicky Sándor. I'm very happy to be invited to moderate or chair this session with actually all these friends. So it's great to be here. So just to position the presentations, I thought of saying a few words about the prototyping fabrication session, which will include 4 different presentations for the next 1 hour. So as we kind of feel here in this room at least, materials that are not standardized or non-uniform or not fully predictable in their behavior is getting increasingly interesting to work with. And in that sense, fabrication and prototyping maybe becomes less of a technical step or stage in our design processes, but rather some kind of new space for negotiation. And this negotiation happens somewhere between our design intent, which I think you will discuss later very deeply, between the design intent and also the material behavior. And these materials, as we experienced, they are not very static. They do change or resist. They shrink, dry. They do many things, I guess we can see on the installations also outside, which make them less of a actors against our planning, but rather participants in this dialogue which we're supposed to have during fabrication and the entire design process, as far as I understand from your abstract. And also very sorry, it's not even my topic of expertise here, but I'm just excited to be with you. So when it becomes this dialogue, I'm very excited to hear about your exploration within these 4 presentations, how this dialogue actually happens, also what kind of tools or technologies you are using in order to support the speaking and listening to these materials, and also how fabrication as well as prototyping becomes a buzzword medium for co-creation for this new European Bauhaus era in our design processes. So with this, I would like to maybe first just list our presenters who are sitting in first row so you can get some eye contact with the audience. We will start with Gonzalo Vaíllo and Lukas Allner. Then we go with Lena Fritsch, Kilian Bauer, and then at the end, Thomas Hamedinger will present, and I will introduce, of course, the paper titles before. So let's start with Gonzalo and Lukas, who actually co-authored a paper, or the research is co-authored together with Karolin Schmidbaur, sitting also here at the back row. So please come to the stage and present unscripted material, vulcanized fiber, and the aliveness of the industrially manufactured in architecture practice.
Yeah, so good morning. We are Gonzalo Vaíllo, Lukas Allner, and Karolin Schmidbaur. Although we right now just present the research or this study, which is called Unscripted Material. Our position is we thought that architectural discourse is increasingly speaking about aliveness, yet often is restricted to living entities, meaning like biotic materials, biological growth, or responsive systems. So with this project, we propose to expand that notion of aliveness and ask whether an industrially manufactured material can be also considered to be alive. And the ideas we showed today are part of our ongoing research project, HAL Forms, which is funded by the big program of the Austrian Science Fund and is developed in the University of Innsbruck. So in contemporary architectural research, the notion of aliveness is often linked to biological systems like in biodesign, self-organization, living materials and entities of responsive morphogenesis and morphogenetic processes. So in this sense, aliveness is typically understood through growth, different sense of metabolisms and living organic adaptation. It is typically understood in the extension of design and construction responsibilities to other living entities, especially or normally plants and animals. And this sense of aliveness, we will argue, is exclusively biocentric. And as a result, what is produced is that the industrially manufactured materials are seen or taken as something dead and static. So they are seen as inner matter, becoming meaningful only when we shaped and instrumentalize and control them under human purposes. So then this is the typical idea of precision in design and construction, which is evident arguably in the modernist dream of total control of the human subject over the construction materials and spaces. But let's say if we take the term aliveness, and we understood it as the capacity to act and transform and affect its surrounding beyond human control and dominance, then we see there the potential to understand also industrial materials as also be alive. Very important, not biologically, but operatively or performatively. And this, if we take this hypothesis, then this will extend the notion of living or being alive beyond the biological towards a material agency, which is actually can be taken as vital. So in our approach, we bridge these two existent strands of work, the previously the two references that we point out, and add material agency through the unpredictability of material behavior. In this case, we take the material of vulcanized fiber, which is industrially produced. It's a natural material that today is largely unknown in architectural applications.
So vulcanized fiber is a material that has been invented in the late 19th century, and it is a predecessor of modern plastics. It has been in industrial production since then, but in the 20th century, it was largely pushed away from the market by fossil fuel-based or petroleum-based plastics, mainly for economic reasons, of course. So a few words about the material itself. Vulcanized fiber is made up of layers of sheets of paper, sourcing cotton linters. That's the very small and short-haired parts of cotton that is usually not is easily usable in the textile industry, so basically from waste. And those are joined into a homogeneous material through a process called parchmentation. In that process, the individual cellulose fibers of the sheets of paper are structurally reorganized in a tighter form of connectivity. So what happens is that you can see this on the top microscopic image. So on the left-hand side, you have the raw paper before the process, and on the right-hand side, you see the parchmented vulcanized fiber. What's happening is that the material is strengthened, and it results in a relatively lightweight but strong material that comes in, that's produced in sheets, as you see in the bottom images from the production line. So it is fully recyclable and biodegradable, since it is not bound with any glue or composite material, but it's only consisting of paper or cellulose. About 100 years ago, the vulcanized fiber for mobility and structural strength was well known. So it was used to make these early versions of shell suitcases, meaning it was the forerunners of today, or was a predecessor of today's aluminum and plastic cases, for example, in that case. So today the material is being rediscovered, and vulcanized fiber is is gaining new attention across different industries. However, today it is mostly used for small, technical, very specific applications, like these sort of washers in electrical fittings and so on. So overall, there is some research, not too much, but as far as we can tell, or what we found, vulcanized fiber is documented, or these sort of works have documented its mechanical properties that suggest structural application in design and architecture, and that's what brought this, besides other aspects, to our attention. Especially when also considering that it is sourced from renewable materials. I mean, cellulose is one of them, or is the most abundant polymer on Earth, and it comes in different forms that can be sourced. So, okay. Overall, so it's relatively strong, and here you see the production process being a wet sort of product in the beginning, and then it hardens in a process. So with all these contexts, the question is, how can an industrial produced material conceived for precision become a site of aliveness? So and that has not been explored yet, but it interests us specifically in this project. So vulcanized fiber is flat, wet, and malleable during a parchmentation process, and when left to dry freely, it will deform in unpredictable ways. So these images you see here at the bottom and also in the video is in a 14-day period. A material that was designed to be predictable, industrial, uniform turns out to possess a performative, almost vital behavior. And in our project, we aim at translating this material agency into productive design methods. In one of our early studies, we worked with assemblies of 9 parts, identical square-shaped sheets, that transform into unique parts in the drying process. And following the same assembly pattern and connection principle, overlapping at the corners, different designs can be created following the same principles. So to compute the range of possible constellations, parts are 3D scanned and aggregated in a digital model, which provides the instructions for physical assemblies. And below you can see 3 different configurations of the same 9 pieces, assembled in different configurations. Later, we moved on to larger scale assemblies, this time with volumetric parts connected by sticks that extend local geometric tangents, so the form logic to the next part. Again, the dynamic behavior, material behavior produces an inventory of unique parts from initially identical wet forms. Like in the previous example, parts are then assembled in a digital model according to a predefined pattern. And then the final form or the final constellation is found in a simulation process. And that simulation process is important because it resolves the discrepancies between, or the differences in between these individual forms and an ideal pattern, the so-called intentional design, if you will. So it's a more of an indirect design process. And here you see the juxtaposition of these 2 processes, the self-forming material itself translated into a design approach that has a similar logic. And then, depending on the part constellation, different possible formations emerge. So it's not an actual design that you can control or design for, but it's more of a possible variation, possible variations, and you get to choose. So what you produce is more of a possibility rather than a goal. So in the middle, you see a photo through a holographic display of an augmented reality headset we used to produce this, so to map the digital back to the physical. And currently, maybe you finish up here.
I mean, this is like the new iteration that we are doing, which a mock-up, it's there outside. Probably to wrap up with what are the conclusions of reflections. In all these projects, aliveness is presented in the participation of the material agency in the design and construction. And again, in this case, it is presented or visible in the discrepancy between the architects or our ideal design as we conceive it at the beginning with particular patterns and forms, and actually the resultant structure. So in this context, we will argue that aliveness cannot be longer confined to the biological life. It's also include the temporal, affective, and transformative capacities of the material. And in this case, industrial materials, if we leave it, if we know how to handle them or given a space for that, can be understood also as alived. And with that, we just wrap up. This is a picture of the image, and then we thank also our sponsors and team along the way for this research. Thank you.
Thank you. Thanks for this great presentation. Now we move on to our second presentation with Lena. Lena Fritsch, please onto stage. The paper or the project which she presents is called Graded Biofoam Systems for Responsive Architecture Assemblies through Robotic Casting. Lena is working at CETA, at the Royal Danish Academy, at the Center for Information Technology and Architecture.
Okay, I hope you can hear me. Yes. Hi, everybody. Thank you for the introduction. Yes, my name is Lena Fritsch. I'm in my last year of my master's studies at the Royal Danish Academy at CETA. Before I studied at the University of Innsbruck. And I'm going to introduce you to my research that I've been conducting since I started my studies there, which is called Biofoam Systems for Responsive Architectural Assemblies. I'm going to walk you through the material system that I developed and also how I use a robot in the end to have as a fabrication medium for that. And also what this means for designing with circular matter. So foam itself is something that we encounter in our everyday life. It's in insulation, it's in furniture, it's in packaging, it's even very crucial for aerospace, and it is valued for being very lightweight, for insulating, flexible, and also super shock absorbing. However great this is, it is one of the least circular materials that we encounter and that we use. The most common types of foam are polyurethane and polystyrene, which are thermosets. That means once they are cured, their molecular structure locks in and they cannot be reshaped or recycled anymore. This means typically that it gets landfilled or incinerated at their end of life stage, and this contributes significantly to toxic waste streams. So the life cycle of a PU foam is a very linear one. So I started to think about how can we rethink that chain and how can we maybe find ways to integrate even the entire life cycle of a material or of this foam into it. And I experimented with a biodegradable and remeltable foam system consisting of industrial byproducts. And I tested out a variety of recipes that can be combined by functionally grading them, using a robot to spatialize these gradients through gravity, motion, and speed. And with this, this initially very linear life cycle turns into a circular one. The base recipe of the biofoam is very simple. It's building upon other research that has been mostly focused on biodegradable packaging. It consists of gelatin, water, glycerin, and a foaming agent. Both gelatin and glycerin are industrial byproducts. Gelatin is a byproduct of the food industry, and glycerin is a byproduct of the biodiesel production. To get it to foam up, I tested two routes. One is called the surfactant-based foaming, which is using sodium lauryl sulfate to stabilize the air inclusions. This is something that is typically in something as simple as dish soap. And the other one is acid-based foaming using sodium bicarbonate and citric acid, which then creates CO2 bubbles. And these produced very different types of foams. The surfactant-based foaming was very homogeneous, as you can see on the top. It produced very stable foams with very fine pores, and it had the capacity to squish and also slowly to recover to its initial shape after squishing it. While the acid-based foaming with the CO2 reaction produced very irregular and coarse pores, which then turned into very lightweight but also rigid foam that after several days of drying became like a very, very rigid structure. So during those studies, I also discovered or observed that all these different foam variants can also fuse together when they are poured sequentially into the same mold, while they are still in a liquid state. So when two mixtures of varying density or viscosity are poured into at the same time, their boundary kind of remains continuous, which means that the material can support internal gradation within a single cast volume, and there's no need for any discrete layering or additional adhesives. So building on that observation, the robotic casting method explored how maybe this unpredictability of the foam while it's still in a liquid stage I can use with a robot, and I explored how gravity and motion can be used to maybe even specialize these gradients. And I used an UR5E robot arm to tilt custom PLA printed molds during the curing, which then enabled a very controlled kind of intermixing of it. So here you can see one of my tests. It showed that lower tilt angles created very subtle transitions, and higher angles, there was very visible layering with very visible flow lines. And also I observed that the timing influenced the degree of gradation. So slower tilting produced very smooth gradients, while faster oscillations made the material migrate further before fully setting in. And I tried out different recipes and different foam mixtures, the stiff and also the very rigid one to combine, and they were allowed to layer in a continuous volume. There was also no delamination and no separate layers afterwards. So this is one of a close-up of the final prototypes that I made, and it showed that the Biofoam has a potential as a circular fabrication medium, because also different batches fuse together seamlessly, and the robotic motion provides a layer of control to specialize these. At their end of life, building upon the property of gelatin, that it can be remelted when introduced to heating, it can just be introduced again into the circular life cycle and be foamed up again. And the robotic casting method showed that both the tilt angle and the motion speed could potentially function as a controllable design parameter and as a grading strategy. So the next step, as this is still ongoing research, would be to quantify these behaviors, measure density, get some data on specific compression and strength, and also finding ways to linking the robotic motion and the robotic casting to a digital data input, which could be curvature maps according to stress or comfort, to link these together. And potential uses for this biofoam could be acoustic panels, adaptive seating, or tactile installations. And yes, I'm very curious to see where this will lead me, and thank you for listening.
Thanks, Lena. By the way, of course, keep your questions in mind. We'll have a little panel at the end of all 4 presentations. So just not to lose time now, I would like Kilian to come to stage. Kilian Bauer will be our 3rd presenter with Material Learnings Embracing Biomaterial Agency in Architectural Education. He is coming to us from the University of Innsbruck, and apology, I forgot to mention, of course, our first presenters were also affiliated with Innsbruck, as they also showed it on the last slide. So, good luck.
Thank you. Hello, my name is Kilian Bauer. I'm a university assistant and PhD student with Professor Coletti at the University of Innsbruck. In my PhD research, which is at RexLab, which is our robotic experimentation laboratory, I focus on the process and material-informed design solutions that enhance the architectural applicability of large-scale 3D printing, with particular interest in the materials, in new materials, and in process inherent aesthetics. Thereby, I often also try to combine teaching and research to give our students a hands-on insight in our work in RexLab. Today I will be presenting the results of a prototyping and design build elective course on the topic of 3D printing with biomaterials, and what we have learned, what my students and also myself, we have learned from working with challenging material systems and addressing the peculiarities of the challenging material by working with the inherent limitations. To provide some background and some context, I would like to briefly highlight the role of RexLab under Professor Coletti, which has a very long history of large-scale 3D printing with various materials from concrete to clay and earth materials and also to plastics. In the recent years, since I have been part of the team now, we have focused to bio-based thermoplastics and more recently to the exciting endeavor of biomaterials. And the term biomaterials here refers to a low-tech, easily biodegradable material family, often on a plant fiber basis like sawdust, mixed with some kind of gelatinous binding agent. Biomaterials promise to be a sustainable alternative to the established materials in additive fabrication. However, while we have been able to do like really large-scale projects with other materials, biomaterials so far only limit us to small-scale objects. And I really hope that we can overcome those limitations by learning better how to work with the material. In our efforts to overcome the challenges of biomaterial fabrication, we figured out two main approaches. One strategy, which is here on the left, would be novel technical solutions, as illustrated here. This is a research task I deal with in my PhD, and I'm very proud that I was given the opportunity to exhibit some of my research on multi-material additive fabrication with biomaterials at this year's BEAM Symposium at FormNext, which is the largest trade fair for additive fabrication, which is also happening this week in Frankfurt. The second approach, which I will talk more about today, is here on the right, and that would be overcoming biomaterial challenges by actually simply better understanding them, learning how to work with them, and embracing the constraints not mere as limitations, but as co-creative design agents. This is exactly what I've challenged my students in this elective course at the University of Innsbruck, and I'm happy to share some results with you here today. First, let's have a closer look at the biomaterial processes for us in RexLab, which actually allow for a fully circular material life cycle, at least for a couple of loops before the material slowly starts to degrade. In the elective course, we started the cycle with old biomaterial 3D prints from a previous research project, which were at the end of their use, here on the image on the top left. Following the material life cycle in clockwise direction, we regranulated the material back into sawdust before restarting the loop with weighing out the ingredients, mixing and bagging it for robotic 3D printing with cartridge presses. The 3D prints are then naturally dried in a custom, well-ventilated shelf for a couple of weeks, and before they reach their final state and ultimately gain their stability. After the material is completely cured, this biomaterial on sawdust basis is surprisingly structurally sound, and the students could assemble their 3D prints in combination with regular construction wood into some really nice prototypes, some of which I will now show in a little bit more detail. I start with Philip, Luisa, and Thomas, which have designed a tile pavilion. They utilize the challenging aspect of the very slow drying biomaterials to their benefit by printing flat sheets on and then let them dry on reusable molds to create shingles in an otherwise unprintable shape. They also went through several iterations on their shingle design, as they learned that the vertical-horizontal cross pattern does not survive the drying process and is a draped position, and therefore the final version of the tiles, which have a diagonal pattern, are hugely influenced and co-designed by the material behavior and the process at use. Next we have Theo, Anton and Laurin, who have designed and built a fully vaulted arch from biomaterials. The hexagonal wedge stones of the arch are thereby both a challenge from and an homage to the non-uniform drying and shrinkage behavior of 3D printed biomaterials. As the moisture sacks down during the drying process, the upper end of a print dries quickly and shrinks more, and unrestricted, while the lower end shrinks slower and less. This challenge made the group go through several iterations to figure out how much angle they actually have to actively design in for their wedge stones, and how much angle will be added on top by the material behavior. Another nice project was done by Lore, René, and Jana, who developed a panel-based exhibition system. They envisioned and evaluated different panel designs and figured out that heavily ondulated, almost spring-shaped print paths, like the one on the top right, give much more leeway during the drying process, in contrast to the print paths with intersections, like on the bottom, which are therefore much more prone to cracking. Lily, Adrian, and Ruben did learn from their 3D printed interpretation of traditional wood joinery that there's a huge discrepancy between the usually very fine tolerances in subtractive woodworking and the much rougher wood 3D printing. They solved these differences by scanning all the 3D prints after drying and shrinking, and then figuring out digitally where to make precision cuts. Leonard, Lewis, and Hermann proposed a project where both 3D printed and regular construction wood are intelligently used in their respective strengths in a small but almost product-ready application of a stool. And the last, a little project by Emma and Vasilisa, who designed their own 3D printed exhibition frames, which I think is a very nice and fitting task for this circular reusable biomaterial, due to the usually very short lifespan of such exhibition constructions. With that, I'm almost at the end. I would like to thank Professor Coletti for providing the infrastructure and the support for this teaching initiative, and of course I have to share all the credits here today for the work presented with all the students who made this presentation possible with their valuable contribution. And I would like to end the presentation with the thoughts I have ended the class, that I hope that my students and myself, and today also some of you, have learned a little bit more how to very generally work with a challenging material system, as it definitely is biomaterial 3D printing. Thank you.
Thank you, Kilian. So thanks a lot for this very inspiring presentation. And with this, we have our last presenter, Oliver Thomas, I didn't know, Hamedinger, who will present a matter of resolution of matter.
Yes, exactly. Oh, perfect. Yeah, hello, my name is Oliver. Thanks so much for being here. I will present to you today an abstract from my ongoing PhD. It's supervised by Brian Cody from the Energy Design Department here at the Angewandte, and Anton Falkais. The title here is a matter of resolution of matter. As the title suggests, the research I'm focusing on is the correlation between matter and resolution on varying scales of the built and architectural environment. The PhD will delve into micro, meso, and macro scales of resolutions, as well as their digital and physical currency in architecture. Today, I only want to discuss the micro scale, focusing on component and facade division scales. Resolution. Resolution is a term often used to describe the number of pixels in the digital realm and is utilized in programs such as Photoshop and rendering softwares to produce mainly digital imagery. Usually it directly affects the time required to visualize all the desired pixels. You can see this when increasing resolution. It also increases the render time, and sometimes working in high resolution can create some sort of a latency between the action and the effect. The term originates from the Latin word resolvere, which means to loosen, to dissolve, or to break into pieces, to reveal more information and data, and therefore more detail, similarly to increasing surface area through subdivisions. This phenomena has been translated into the digital realm and fed back into reality through AR and simulation tools like Karamba, for example, where we can accurately predict how materials and structures will behave in reality. However, in fabrication workflows, this is not always the case. As we have digitalized the way we work in architecture, mathematics has been at the backbone, even when we draw splines and create NURBS. Behind the scenes, the computer is calculating how that even works and how this is possible. The tools we use to fabricate often don't really align with how we design digitally. We might simulate behavior in voxel-based models, then smooth it and force it into a continuous toolpath again, which strips away much of that digital intelligence. The result, texture and resolutions, are more closely dedicated by the fabrication process than by the design intention. You see this even clearer in FDM 3D printing. Our simulations are still crude, the material behavior and setting times vary, and we can't reliably predict performance, especially for tall or long-lasting structures. So the real question is, can we develop fabrication tools that can carry this digital syntax all the way from simulations to physical outcome, without losing or potentially gaining data in translation? The binder jet printing technique offers a direct path here to align digital thinking with digital fabrication using a voxel to matter procedure. Philippe Morel identifies this latency and inconsistent computational workflow. He addresses them through a series of computational chairs, a functional object, yet whose form is derived slowly from computer simulations. They are testable, automatable, and reproducible, introducing the digital discrete and voxel-based workflow that materializes digital resolution directly. Contrary to this, ornament is often framed as a surplus, an additive layer applied for appearances and yet closely related to increased surface area and therefore to the resolution. I want to question whether that surplus material on facades can become integral to performance and repurposes an ornament as an operational feature, so where ornamentation shifts from a mathematical aesthetic add-on to an operational element. The use of ornamentation and its application have developed alongside society's emerging trends and culture and economic shifts, appearing slightly different in each of the periods. On the left, the Giralda is hand-carved relief. Then we have the Ravensbourg College. It's a paneling, an early example of digital design and parametric tools. Then the City Gate in Malta from Renzo Piano Architects, which shifts towards tectonic articulation. And last but not least, the Digital Grotesque from Hansmeier, which illustrates the digital detail that is impossible to reproduce by hand. My claim is not that one replaces the other, but we can now steer ornamentation toward operation, so away from an add-on style and toward surface difference that support performance. To talk and evaluate that shift, I'm using this 4-quadrant diagram. On the x-axis, we map value from aesthetic to performance, and on the y-axis, we map cost from craft to data. The Giralda sits in Q3, which is a combination of aesthetics and craft. High visual value made through a lot of handwork. On the performance side, the City Gate in Malta is very interesting because it would sit in Q4, which is performance and craft. The articulation provides self-shading, which gives a measurable thermal effect, but is still heavy stonework and requires skilled craftsmanship, which increases the cost. On the other hand, the Digital Grotesque lands in Q2, aesthetic plus data. The surface is highly complex, yet additive fabrication makes voids cheap and complexity manageable. So the cost would shift or move towards data, not to craft hours. So across these cases, we can see aesthetic and data on the one side and performance and craft on the other. The still empty quadrant from before, we noticed, in the diagram is where performance and data would align. And this is where my work aims to create a moment when ornament becomes functional, when value arises from measurable effects and costs scale towards resolution parameters rather than manual labor. Here's how this idea could be implemented or shown on a facade. The blue marks the water channels. It's a vertical micro-relief that guides the drain to the drainage outlets. We keep the historic ornaments in place, and we add integrated planters at ground or window height so they are easy to service. The green marks the bioreceptive surfaces, which is a microtexture intended to host moss and small species, not as a decoration, but rather as a function. To explore this hypothesis, physical prototyping was utilized. A total of 25 3D sand-printed samples were developed, designed, and fabricated using the binder jet and voxel-to-voxel fabrication techniques. These samples were designed to have a certain level of details. On the left now we see the sample family. They consist out of LOD1, LOD0, LOD1, and LOD2. Each of these LODs have a different articulation of the surface. We start with a complete smooth surface, then a high articulated surface, and then a very complex surface. On the bottom right is the operational scale graph, which maps the effectiveness of the surface articulation in relation to the height and resolution of the ornamentation. To check if these different surfaces have an impact directly linked to the fabrication scale, the grain height, and the boundary limitations, I ran bench lab environmental tests under controlled conditions. Here we can see some of them. Mostly they were consisting out of a water erosion test, a UV exposure test, a freeze-thaw circle, and a water absorption tests. All of these tests have been performed to align as much as possible with the relevant industrial standards. So, for example, the freeze-thaw circle was a combination of 24 hours in a water basin and 24 hours by minus 15 degrees over the period of a whole month. So this research aims to shed light on methods that could help to cool urban environments with low-tech smart facades and reconnect buildings with the surrounding ecologies, treating resolution not as an aesthetic choice, but as a performative parameter that shapes the spatial expressions and environmental responsiveness. Through the synthesis of theoretical inquiry and material practice, this project proposes a model of architecture which is materially specific, ecologically informed, and critically engaged with the complexities of contemporary designs. Rather than dismissing ornament and surface complexity as decorative, the research positions them as a critical driver of circularity and design intention. Thanks so much.
Thanks. Don't go far. All right, thanks for all the presentations. I think they discover or they go around quite interestingly the same or similar topics. And before I ask my very selfish questions, I would like to know if anyone from the audience has anything to ask from any of the projects or from overall. Oh, okay. So then maybe I can start second row.
I have a question for the vulcanized fibers. You mentioned that you use textile waste, but use only cellulose fibers. And in textile waste, it's often the problem that it's like mixed with some polyester sewings or whatever. So is there a little percentage of synthetic fibers inside, or
Thank you for the question. So maybe to put, I'm not sure if this was clear. So the material we are not producing ourselves. This is like an industrial product that has been around for a very long time. And as far as we understand the production line of the product, it can vary in between sourcing paper or cotton or a mixture of the two, dependent on the specific technical applications that are required. But since it's also a production line that predates the existence of petroleum-based materials, they are very keen on, I mean, they can't use synthetic materials, otherwise it would contaminate or, disturb the product. So as far as we know, it's purely cotton, or it's essentially cellulose, but it can be from cotton or paper or a mixture of the two.
and is there then a life cycle? So can it be
It is.
reused, or?
It can be. So these these forms can be re-wetted or reformed, but not exactly recycled in the exact. So you cannot shred the material and reform the same, but the material is biodegradable. So I guess that's the sort of trade-off for the increased strength in comparison to paper, which is recyclable. But since it's bonded only mechanically on a macroscopic level, this material is sort of welded, so you can't rip it apart and recycle.
Thank you.
I have a question for Lena Fritsch. The foam is partly made out of gelatin. Is that based on animal protein, and is that a waste product? And what is your take on that?
Yeah, it is a byproduct. Gelatin is mostly a waste product from the food industry, so from pork bones or like all sorts of animal bones. I delved into the route of experimenting with agar-agar-based foams, and that's what I wanted to experiment more. However, these could never reach the density and the kind of strength and just properties that conventional foams, as I say, has, which is then why I delved into gelatin-based foams. And since it's a byproduct, since it's something that would either be wasted or used for other kind of commercial produces, I deemed it to be okay for my take. Yeah, so
definitely.
I think it was okay.
Thank you very much.
Karolin? Sorry, Petra had first, and maybe then Karolin.
Thank you. I have another question for the foaming process. So you used a robot to define the tilting angles, but you still did a manual injection of the material. So I guess that's not exact enough to be really repetitive as a process. Are you planning to alleviate that in the future by having maybe 2 robots, or I don't know, changing this process to be a more repetitive system?
Yeah, exactly. That is a very crucial next step that I want to explore more, which is using another robot to inject the specific foams. At this early stage, as I call it, mainly I wanted to find out if it works, if there's any delamination or how these two could fuse together. So it was kind of a human-robot collaboration in that sense. But in the future, as I want to delve deeper into maybe having curvature maps or performance maps, which can then be applied to these gradients, I need to be more specific. So that's where I want to explore how a second robot could maybe come into play with that. I discovered, I tried 2 different routes. Once where while the robot was moving, I was injecting the foam, and once where I was injecting it, stepping away and letting the robot do its thing. But yeah, in the future, I would like to automate this process even more.
Thank you. I have a follow-up question. Did you experiment with yeast?
No, I haven't. Not yet.
So that would maybe also be a great role model for biological foams.
Yeah, thank you. We'll look into it.
So we have a question from Karolin from the back, and then we will also, I think we'll have 2 more.
Yeah, thank you. Just a quick comment, not a question. I wanted to answer to the question or offer another bit to the question of the recyclability of vulcanized fiber. It is fully recyclable because it could just go into paper waste and then be treated just like paper waste into a new paper and into a new vulcanized fiber. So it's completely cellulose, either from cotton or from wood, or plant material, but it is fully recyclable in that sense, just like paper. No treatment of the waste.
Karolin is also co-authoring the paper from the first presentation. Yes, we have 1 more question here.
Yes, another question for Lena Fritsch. I'm from the physical department at Bodenkultur, so a little bit more on the technical side here. Did you use other curing methods for the foams? Did you like try stirring or shaking? Because with the robot it looks more very arbitrary and very out of the common, because some of you use like stirring or shaking for mixing. So did you try other methods too?
There is currently another project at CETA, or in collaboration with CETA, that is called rotoforming. It is like the robot kind of has a mold as an enclosure and then forms it. So because right now I have an open-faced mold, as I call it. And I'm currently in the process of exploring that further. But so far, the things I have explored was different tilt angles and different speed parameters. So I went from, I think the lowest was around 10% of speed of the robot, and the highest was like the full 100%. But the rotoforming was something that I am kind of in the process of maybe developing that further as well. I hope that answers it.
Yeah, thank you.
Any other questions, maybe? Thank you.
I have one question which would address, I think, almost all of the presenters. You're all using digital design and fabrication processes, and the material is the use of material is very experimental and alive, as you called it in the beginning. I would be very interested which methods you're using to bring back this data you're monitoring of the experiments into the digital process.
Let's start on this side now.
Okay, sorry, what was the question again? For real, what exactly was the question?
The question was, as the outcome of your experimentations is very experimental, and you, I guess, are monitoring those sets, how would you bring back this data into your digital process?
Okay.
So what is the feedback?
Yeah, I mean, the feedback is quite literally, it is like you, as in my project, it always works with the same. So we design on a voxel based, then we fabricate, which is also on a voxel or a bitmap based, and then we measure again. For example, we measure with thermal imageries, which should have the same resolution than the fabrication, so we can feedback then exactly what we notice in the tests. So we learn directly, and then we can adjust our simulations, because the design itself from my project is not based on specific design intents from my side. It's based on, for example, maximizing surface area to create most self-shading, and that's then feedbacks directly into the simulation again. So it's like, it should be a closed loop in a way.
Yeah, working with biomaterials, maybe it's a little bit less precise. A lot of the feedback is more like experience and trial and error, and just figuring out what is possible with this material. One approach I had in my presentation was like to afterwards scan the material, bring it back into the digital. I think that's also something guys over here you also do, but also to some extent live with the unpredictability.
Just one quick note. I think my biggest learning was that there's not 100% predictability, and there's a lot of properties of these biomaterials that might not always work in our favor. And I think what I learned from it is that I can make these properties kind of approach as my design intends. For example, I think my biggest struggle at first was the liquid state of the biofoams, because I didn't know how to mold these or how to actually use this as a fabrication medium. And then that's when I got into the robotic casting method, because I thought maybe I could use this unpredictability of the foam in a liquid state to kind of make this as my use. And yeah, I agree, you cannot be 100% exact, but maybe for specific use cases, you don't need to. You can be as exact as possible. But yeah, I think it's very exciting to also design with this sort of unpredictability and have it as a parameter.
Thank you. For us, this question of translatability in between digital and physical is quite crucial, actually, to the process. So I think at the threshold, I guess for us, there's mainly 3D scanning involved. So to capture first the material state in its different form as a geometry, and then on the other end, we briefly showed this was we are using augmented reality to overlay the digital information back to the physical in order to build the structure, and then then again it comes back to the digital through scanning to compare, yet another cycle of unpredictable effects that occur from the construction process, from the material behaving under stress, sitting, and so on. And we use that information brought back to the digital to leverage the possibilities of control and uncontrol in the design process. So it's a sort of back and forth in between.
Thank you. One last question. There are two. 1 second. We might allow this. I am looking at the organizers. Yes, please.
It's kind of for all of you. I haven't taken note on who mentioned the word aliveness, but I think quite a few of you have. And I was just wondering on your definition of aliveness in context of your material that you're working with or your process and how you're defining it. Because I think it has been used very differently from talk to talk.
In our case, is actually the whole argument of the presentation trying to extend the notion of aliveness to not just living organisms as we understand them, but also as a provocation, saying that this is also in what we initially understood is completely as the inner matter, which is industrially produced. And so in this case, we define aliveness in a very post-anthropocentric manner, in the sense that it's all that is beyond the human control and precision, at least in the material. So wherever the material behavior surprise, affects, it doesn't need to be good, it doesn't need to be bad, because this set of values, whether it's good or what is bad, actually doesn't fit. It is an ongoing evaluation. And in this sense, aliveness is, to put it simple, everything that is outside human precision and predictability.
In case no one wants to jump on this answer, we have one more comment here. First row, very ambitious.
Thank you, Vicky. I want to jump on the topic of aliveness and feedback. And I believe in all the kind of processes or growth processes in biology, feedback is essentially built in as a real-time effect, right? And I don't really see it in all of your presentations yet. So I think to achieve more aliveness in our cultivation methods, this is also a really important principle to embed into the generation and processing schedule, so to speak, to have this kind of real-time sensing and reacting and adapting. And I think this could be an interesting thought for all of you to, think about more what could be the next step to make it more alive. That's just a comment. Thank you.
Thanks a lot. I'm actually very excited about this topic of how we communicate. Is it a synchronous communication between you and materials, or something way more with delay, asynchronously? And if the synchronity is something temporary, so do we just test this way? So on the long term, we actually try to be more automated or programmed with these materials, or this is a new way of designing or a new attitude towards materials. So I find all the papers very exciting. It's again not really my topic, but I really enjoyed all the presentations, and I think with this we could close this session and go for another coffee and food break, if I'm correct. So thanks a lot for everyone for the questions, and enjoy the day.
All right, I'm going to start. Welcome back, everybody, from lunch. We are starting now with the panel Bioreceptivity. That's the last, before the very last panel of today's symposium. My name is Barbara Imhof. I'm a professor of integrative design at the University of Innsbruck, and we focus on extremes, meaning we look at reciprocities between Earth and space architecture and design. That has to do with my other affiliation of Liquifer, where I'm co-leading a company which has to do with space architecture. And I was just thinking, bioreceptivity is really about materials that sort of are welcoming bios, life, and what you want to do in space. For example, at the International Space University is the opposite. You want to keep any other life than human rather out, because it's very difficult to control because you have a very limited biosphere and life support systems available. But what we do here is the opposite. We sort of welcome everything which is alive, and we also design for this. And in this panel of bioreceptivity, we especially look at membranes, at sort of the interfaces between the inside and the outside, the facades, and how these kind of barriers can become more translucent or permeable through different specific designs of materials, but also observation of different materials which already grow on it. So bioreceptivity is, as a definition, is the ability of a material to be colonized by living organisms. And these organisms, they can include fungi or algae or tardigrades or microbes, of course, lichen, any kind of these kind of organisms. And that is a term which originates in the fields of building ecology and conservation. And it's interesting that architecture has, in the last, I would say, 15 years or so, really have been extending to other fields and trying to bridge and also integrating other disciplines. And that's also what we can see here in the symposium, because a lot of projects, research is informed by these other disciplines and requires a very close collaboration. So it's sort of an extension of the architectural field into these other realms. What we will hear now in this session are 4 presentations of different kinds. They will all look at these kind of membranes, at sort of bioreceptive materials, but also at the organisms and also at building systems, and how these building systems, for example, the grey water, can be used to also feed the sort of the living architecture on these interfaces. So what we do is there's one recording, because the first presenter, Sneham Pandey, he couldn't come due to visa issues, so we'll see a video, and then we'll have 3 presentations, and we do it as we did with the other sessions. We have the panel afterwards here, and I'd just like to would like to start and would like to welcome the first presenter who is not here. But I think he still deserves an applause, probably. So it's Sneham Pandey. He's from the IAC in Barcelona. It's his PhD work, and it's titled BioDiverseCity, Bioreceptive Surfaces for Urban Ecologies.
Hi, good afternoon, everyone. This is Sneham Pandey. I'm joining in from India, and I'll be presenting my research on bioreceptive surfaces. This project was done over the course of 6 months and was part of my master's thesis at IAC that was finished last year. And the seed or the spore of this research started from an article by Madhusudan Katti, where he talks about that unlike coral reef cities are not fountains of biological diversity, they weren't really built for anyone else other than humans, but still a lot of biodiversity seems to thrive within our cities. And he asks this question, what would happen if we were to consciously design our cities for multiple species interaction? That led me to my core research question, which was, could bioreceptive surfaces aid in ecological connectivity, and how can they enhance urban biodiversity? As we know that urbanization leads to the fragmentation of nature into isolated fragments, and you know that there are physical and chemical barriers that inhibit the biological activity of organisms. But then under this umbrella term of reconciliation ecology, there is research that suggests that why don't we use urban surfaces as ecological surfaces that could either act as small or medium-sized habitat patches, or these stepping stones that could enable the movement of species across fragmented landscapes. Well, that remains the main research question. In this few months that I had, I was able to focus mainly on the fabrication part of the research, where there were 4 phases to this question, where I looked into the client or the organism that I wanted to work with, then the host or the scaffold material where this organism could thrive, design morphology to give space to this organism, and the tool or the fabrication method that would enable this design. In terms of the client, I picked moss because it has this multiple ecosystem services that it provides to a city. So the initial phase was just understanding the requirements of the client, what kind of pH, what kind of humidity, what kind of spaces does it like, and also trying to see the food web that's connected to moss. So to understand what other species does it help. So mainly moss has a lot of insects that grow that feed on it or take shelter in it. And it's also indirectly connected to birds, because these birds then feed on those insects, and birds also use moss as a nesting material. In terms of the host, I looked into the intrinsic factors that guided this research, where porosity, roughness, the pH, and the color of the material was an important element. But as an added layer, I also wanted to prioritize materials which are bio-based, which are waste-derived, and they're locally available. So based on the literature review, there were multiple materials that I initially tested with, such as eggshells, sawdust, charcoal, cork, coffee, and mussel shells, even clay. But through the course of the research, I decided to pick cork and charcoal, both being wood-based materials, which is something very special to the course that I study, because we work with carpentry a lot. Cork is mainly used because it's porous and a fibrous material, and it's durable, and it also provides thermal and acoustic insulation. It's quite popular within Spain and Portugal and is also been used as a cladding panel, and it comes as a waste product from the cork industry or the wine industry, and so that itself is a great material choice. And charcoal was used again because it's locally available material. It's acidic. It's used in gardens to support plant growth. It also absorbs water and retains humidity for quite a long duration. Uh, in terms of the design, the intention was to create a surface which would have a large surface area. It would have these varied porosity and humidity pockets, and the design should also be in a way that it enables self-shading and water retention. So based on the material choice, that was cork and charcoal, the prototype was designed as a cladding panel. It's an interlocking modular cladding panel of the standard size of 1 meter by half a meter. The surface geometry was picked as sine wave modulation because it has these alternating crests and troughs, which allow a varied amount of humidity and also a large surface area. So there is this solar exposed part, and there is also these subtle water channels that could allow the flow of water and allow more moisture or humidity to be present on the surface. And the design was also given a 5-degree slope so that it slows the movement of water and allows more water retention. Coming to the most exciting part of the prototype, which was creating this functionally graded biocomposite. So the Ladybug tool was used as this logic for segregating the materials, where the solar exposed parts of the geometry were supposed to be printed in cork, and this more subtle self-shaded parts or the water channels were to be printed in charcoal. And all this was possible through this technique called multi-material additive manufacturing, and this very specific end effector that was developed by Ricardo, Riaz, and Nestor in this workshop that I happened to attend at EAC. And through this process, we were able to come up with a product which is simultaneously that simultaneously prints two materials at the same time. So we are able to print a material which has cork on the solar exposed parts, and it takes care of the insulation, and then charcoal in these water channels, which could aid bioreceptivity. So in terms of the application of this specific prototype, it was designed to cater to this existing demand for retrofit buildings in Barcelona, where the city council and the housing consortium are providing grants to people for refurbishing their houses in an attempt to reduce energy cost so that they don't have to really destroy the building. They can retain it as it is and just spend money on the renovation. And also Barcelona has a lot of research done by the environment department where they suggest that the city has huge amount of area that's available for green roofs, and also a lot of area that's available for vertical walls. So potentially that surface can be tapped into and be used for bioreceptive surfaces. In terms of the immediate results of the prototype, there was about 10% shrinkage, and so that's something very important to understand, I guess, as when we print with biocomposites. Also, there was a drawback to this research where we did not use very fine core granules. But if we were to use another material which is finer, we could have more detailed prints and go for a morphology which is even more detailed and intricate. As the next steps, there's still a lot of questions that are left to be answered, but specific to this fabrication, I also realized that Barcelona is not the most humid place and it remains dry for a large amount of time. And there are certain researchers where they looked into this layered porosity um, of varying porosities, which could allow for better water retention for moss. So that's something to be tried maybe in the next phases of this research. And also, I looked into gyroids, and growth simulation patterns, so that they could be explored for multiple species interactions because they have this geometry which is so varied, and it will have a microclimate which could not just cater to moss but many other um, species. And these two have already been used in bioreceptive researches, but it'd be so interesting to look into this particular technique, the multi-material additive manufacturing, and then create these geometries with two different materials. In terms of the bigger scheme of things or the urban element of this research, the research could also look into how could we map this bioreceptive potential across urban vertical surfaces, because maybe not every wall would have the requirements that are necessary for moss or another species to grow. And also, it would be really interesting to look into what additional urban elements, such as benches or fences, could we use? What other materials could be used in a similar pattern that could aid biodiversity? For example, mussel shells could have also been a great material choice for Barcelona, given the amount of seafood that it generates. That said, thank you so much for your time, and thank you for giving me this opportunity to present remotely. If you have any other questions, please reach out to me on my email.
Apologies for getting sort of saying he instead of she, some misunderstanding. We'll go on to the next presentation. Unfortunately, Sneham cannot be here. It would have been nice to speak a little bit later about her work. We go to the next presentation by Alexandra. She's Alexandra Lakatushu. She's from the BioID at the Bartlett. There's always a lot of exchange, I think, between on these conferences. And she will be presenting her PhD work, which is going to be finishing early next year, hopefully, right? About moss and the study and the observation of moss and the monitoring of moss. Presentation is called When Moss Speaks, Informing Bioreceptive Design Through Mapping the Photosynthetic Behavior of epolithic mosses. Good. It's with Markus Cruz and Brenda Parker. Thank you.
Thank you very much for the introduction. Can you hear me? It's really wonderful to be here in the company of such fantastic people doing such great work. It's really exciting. Thank you for having me. Yes, the title. My name is Alex. I am currently a PhD student researcher at the Bartlett School of Architecture, studying under Markus Cruz and Brenda Parker in the BioID group, which is a mix between the Bartlett School of Architecture and the Department of Biochemical Engineering at UCL. Today I will be presenting a part, not a very small part, but quite a small compared to all the work I've been doing lately of the PhD. Okay. So my work, my research is at the intersection between, well, my background is architecture. I think I need to start with that. And throughout this research, actually, I have moved a little bit more into scientific research, which is where the PhD took me. I never really intended to do all of the things that I've been doing, but it's been exciting nonetheless. So my work sits at the intersection between laboratory experimentation, hardware and software engineering, the design and building of bespoke bioreactors to test materials, but actually most of all to understand mosses, how they grow, and how we could foster them better within our built environments. Biocolonization of building materials is a very common and natural process that happens all around cities and not only. Mosses are essentially kind of microforests because they host a wide variety of microorganisms, and they also participate in many ways within urban ecologies. Sorry. Where am I going? Sorry. But the problem is that actually when it comes to bioreceptivity and kind of encouraging the growth of organisms on buildings, we actually don't understand very well what is happening, and we cannot really control what is happening. So part of my work was actually to untangle some of the very entangled relationships in moss growth, and actually in doing this with the aim of accelerating growth on building materials. What you see here is part of Marcus Cruz's ongoing research for the last 10 years. He has been developing bioreceptive concrete materials and testing them at different scales, at smaller scales, but also at larger scales, such as this panel here. And this is one of the prototypes that we have well, it's not exhibited, it's actually in use at St. Anne's Catholic Primary School in London, where we have transplanted mosses quite a few years ago and have monitored their development in time. And spoiler alert, it did not work. So we went back to the drawing board and started thinking about, right, how do we actually understand them better? And how do we design for them, starting from the organism up? One of the biggest questions I get all the time is why moss? Well, just to put things in a bit of a perspective, moss is about 400 million years ago, where carbon levels were between 14 to 22 times the current levels, and the temperature was 4.4 to 17.5 degrees Celsius higher. They migrated to land, and when they did that, they absorbed so much carbon out of the atmosphere that they triggered the first ice age. So they are very minute organisms, but incredibly powerful. So understanding them would be really important for what we're trying to do in terms of climate change and kind of living with climate change. Yeah, so other than that, mosses also are, if we look at it from an instrumentalist perspective, mosses are fantastic ecosystem engineers. This is also because they produce soil, because they absorb a lot of nutrients from the air and deposit them. They also capture particulate matter, which also contributes to that soil formation, but also they capture water in a matter of minutes, large quantities of water, up to 15 times their weight in water. So the benefits are clear and they're out there. Along with that is biodiversity and so many other, including temperature, regulation of surfaces. But in order to actually grow them on or accelerate their growth on buildings, we have to look at an ecosystem level, or at least a micro ecosystem level, because part of how mosses colonize buildings has to do with material bioreceptivity, of course, so the properties that materials have that allow for colonization to happen. That could include pH, it could include chemical composition, it could include leaching patterns, but then that's not the whole story. So the other and most important part of the story is the environmental conditions, and those include light intensity, water availability, and obviously temperature as a kind of driver of drying. And of course, if we want to understand them better, we need to understand their ecophysiology, so the relationship that they form with materials, with environmental conditions, but also with other associated microorganisms that sometimes we realize that they might actually be the architects of moss. And when it comes to growth, we have to understand how they grow, and how they grow is through photosynthetic activity. And that is very much reliant on water. As I said before, mosses absorb water very, very fast. And when they do so, they activate their photosynthetic tissues, so the cells that contain chloroplasts, and they start photosynthesizing. And then as soon as they dry, they will stop photosynthetic activity, and they will actually start losing carbon from their tissues. So that is a very, very important dynamic that needs to be taken into account. So carbon, on the one hand, is a parameter that we care about because of what mosses can do for us, but can also be considered a proxy for understanding what the optimal conditions might be for their growth. So part of my research was to actually understand this dynamic, and to understand this dynamic, I have built an instrument in order to monitor in potentially in near real time, but actually experiments that I'm doing are not the data stream is not that fast, but we are essentially monitoring things like carbon dioxide, temperature and humidity, light intensity and color, and oxygen levels. And this is a closed loop system where we have the specimen, we have all of these sensors that I mentioned, and also the moss and a camera to also monitor the light reflected, so essentially image analysis, so the light reflected from the tissue, the sample tissues, and that is in the near infrared spectrum as well as the visible spectrum. Yeah, so the protocol is essentially we have a measurement every 15 minutes over 3 to 4 weeks, and then we analyze the data. And what the data shows us is a behavior in time of carbon retention and release, but also the relationship between day-night cycles and photosynthetic activity. And then we can see in the image right there the wet sample, which is more kind of green coloration, and then the dry sample. So we can also see where exactly in the specimen photosynthesis might happen. And that might is most of the time very much related with the water contents. Before I kind of end, I wanted to say I really love this quote by Ursula Le Guin. Do you realize, the phytolinguist will say to the aesthetic critic, that they couldn't even speak eggplant, and they will smile at our ignorance as they pick up their rucksacks and hike on up to read the newly deciphered lyrics of the lichen on the north face of Pikes Peak. So I guess my question in this research, more and more I realize, is can we speak moss? And if so, how can we learn to speak moss? And can data have some sort of impact in this? Can we use data to learn how to speak moss? And also, how would this influence design looking forward? And this is a bit of a short video and then the last short video I will show at the end. Hopefully this works. Oh. So what is happening is that we are, the data, that is the carbon dioxide and the water content, are being sonified, as well as the light intensity when it is present. And then on the left-hand side, we can see the specimen with the varying levels of photosynthetic activity happening throughout time as the moss dries out. And with this, I will leave you with my last video, which is kind of just wrapping up all I have done in this, and then looking forward to the conversation later.
Thank you very much for the really nice imagery and the wonderful presentation. Now the next speaker will talk later about it, more about it. The next speaker is Nathaniel Myers. We stay in London, we stay with the BioID. Please come stay. It's the presentation is titled Weathering as an Ecological Design Factor. So it's another topic, but we stay with the interface, with the vertical envelope of the building, an exploration into biogeomorphic interactions for enhanced weathering of urban stone structures. And your co-authors are Maria Jose Creyas and Farida Radwan.
Yes, thank you. It's good to be with you all today. As Barbara kindly mentioned, I am from BioID, and speaking on behalf of my co-authors, Maria Jose Cujedes and Farida Radwan, with the support of some amazing tutors, Andreas Korn, Annette Salome, and Tony Lee. Central to our project, we ask how intentional weathering can generate young soil within the built environment, using functional ornamentation as a mediator for regenerative bioreceptive design. Weathering is normally understood as decay, something to defend against or power wash remnants off of, but geologically, it's at the core to our planetary engine, unlocking nutrients, regulating CO2, and contributing to soil formation. Building off of the urban cliff theory and the premise of enhanced rock weathering in agricultural practices, building facades and associative material in turn become fertilizers to their surrounding environments. Embracing the phenomena's transformations across time, weathering is reconsidered not as a detriment, but as an emergent aesthetic and ecological function, an opportunity to design with. Central to our research is the question, can we design a methodology that creates that creates a self-sustaining and resilient urban ecology through the built environment. We explore biogeochemical nutrient cycles activated by weathering and use them to intentionally guide biological growth on urban stone across time and scales. This reframes the facade as a material and a functional interface. Our work connects the geological processes with observable urban weathering, focusing on 3 premises: pioneering microbes, such as moss, in this case it's lichen, spontaneous vegetation, and water as a vehicle, developing strategies to foster these weathering agents so, in turn, that they initiate ecological succession and a cadence to produce young soil. We began by surveying weathering on limestone buildings across London. Patterns of runoff, pooling, greening, and higher plant species then informed our design hypothesis. In contrast, we went to more rural lithic environments, recognizing that London, through the Great Fire that happened in 1666, was primarily sourced from a particular quarry down in the south of England. There we did a stereotomy exercise, and it was interesting to compare the full spectrum of lithic environments untapped, where weathering is a constant. Mapping became an essential exercise, and we kept returning to it to help us delineate not only our approach within design, but also all the different information pathways. Within this, mapping also highlighted successional activation through interrelational forces. This was important to recognize that these are dynamic systems, and they are constantly at play, spanning across scales, nutrient exchanges, and time. Central for us was the nutrient of phosphorus, essential for plant growth, and as a macronutrient, it's now deemed as a silent global crisis, termed as the phosphageddon. We leveraged microbial weathering of lithic colonizing lichen to mobilize inorganic phosphorus, and through their metabolism, making it bioavailable in the form of phosphates for the higher plant species to uptake. Lichen, this is the area that I tended to focus on within this project. Lichen or microbial mutualism partnership between a fungi and a photosynthesizing partner. This symbiosis is resilient, self-sustaining, and it enables the ability to colonize harsh environments such as bare stone, deeming them as a pioneering organism, agents of transformation through their metabolic chemical interactions. Lichens weather stone by secreting organic acids. We tracked the acidification and the role in unlocking phosphorus through phosphorus screening mediums, demonstrating that at the microscale interaction can drive a macro-scale greening through soil and root mechanics. We continued the research promising partnership through CO2 recycling, co-cultures of Aspergillus niger, and photobionts of chlorella and nostoc. My co-author Maria, she helmed the plant interaction. There we go. As research spans across time, we needed to track the seasonal rhythms of London's ecosystems, considering the microbial activity, moisture, sunlight, and interlocked life cycles of these higher plant species. In parallel, we explored lithic plants native to England, which naturally grew on highly alkaline surfaces. We developed a soilless substrate that, from biodegradable wastes that support seed establishment and microbial interaction. Another research pathway helmed by Farida, water was the main carrier of this weathering. Our study focused on capillary rise where water ascends through porous stone, generating gradients of moisture and pH, allowing for microclimate diversity needed for multi-species inhabitation. Geometry strongly shaped this water movement. Niches, corners, and shaded recesses all hold moisture longer, assisting the formation of these ideal microclimates while acting as the transport pathways for different nutrients. This became a core parameter in designing across the research avenues. We extracted the geometric language of London's neoclassical architecture, analyzing the profiles, joints, and rustication, reinterpreting them as a functional opportunity rather than a purely decorative cultural expression, combining weathering agents across viability specificities. We tested various calcareous materials and prototyped geometries to visualize how moisture rises and persists. Weathered prototypes reinforced that deep pocketing supported seed establishment and exposed surfaces situated microbial biomass formation. The result was a functionally differentiated motifs. We evaluated each profile's interaction with light, wind, and water, mediators for producing diverse ecological niches. By orienting and combining these profiles, surface articulation could strategically create conditions that favor synthetic lichen, seed propagation, and moisture retention for new functional expressions. We tested this with a chair prototype 1-to-1 scale that integrated the profile, stepping, and ornamentation language. The prototype is an active experiment currently in outdoors in Waterloo, London. Rough, textured, and articulated surfaces correlated strongly with bioreceptivity. Formalizing this relationship, ornament became a mediator accelerating weathering and in turn integrating structural greening. Using rainfall and solar simulations, we placed different coal cultures where the conditions best supported them, similar to the strategies of environmental profile analysis that we did, creating a seasonal, responsive, and biologically considered inoculation approach. We then applied this design logic within a site intervention in Trafalgar Square. It's very intensely maintained limestone and densely touristic. There, through spatial analysis, we identified areas proposing a new seating intervention that could integrate ecological function, contrasting the typical building maintenance while fostering the local ecology. The resulting structure merges profile reconstruction with stepped channeling witnessed through our different case studies. Embracing weathering as a dynamic process, this systematic design approach integrates life cycles, associative nutrient fluxes of both decay and growth into an architectural expression. Modern buildings have been stripped from material thickness, roughness, and ornament. However, beyond the decorative, integrated ornament can enhance biological functions of a structure, promoting the connections between building and the bioscene. Thank you.
Thank you, Nathaniel, for the really interesting talk. I like these facades and how they sort of sometimes shape into something else, into something living. We come to the last talk in this round, Dimitra. She's gonna talk about water, great water. Welcome, please. Dimitra Albani-Leka. She's from the University in Ioannina, and it's her PhD research work under Yannis Savoleas. And it's also, this research work is also part of the Excellence Cluster Matter of Activity, which is led by the Humboldt Universität. The title is Water-Driven Membranes, Methods of Activating the Building Envelope for More Than Human Commoning. Welcome.
Thank you. So, hi, my name is Dimitra Albani-Leka. It's okay? Good. I'm a PhD candidate at the Architecture School of the University of Ioannina, and I'm a researcher at the Cluster of Excellence Matters of Activity in Berlin. I also work as a landscape architect in Berlin in parallel. I have been a member of the MycoX collective and have worked extensively with fungi in different projects. Today I will not speak about mycelium, although I was excited yesterday to hear all those talks. But my former investigation is relevant to my current work that eventually has to do with the quote from Lars Blank: "You might want to ask what microbes can do for you, but you also might want to ask what you can do for the microbes." I am largely interested in learning from organisms and their temporalities, and what conditions they prefer in order to thrive, and how I or we as architectural designers can program and promote their growth and harvest their or tap into their activities. My presentation today is more methodological and touches upon design thinking that forms my thesis. I investigate methods of activating the building envelope with grey water in order to promote biological growth and support urban biodiversity. I investigate the correlation between water harvesting, circulating, absorption, and accumulation morphologies and materialities, and the growth of microbial biofilms and plants like mosses, and in a circular thermodynamic chain reaction, how those small organisms can create the conditions for urban fauna to thrive. In my thesis, I see the building envelope as a potentially active membrane that hosts an intermediate new type of space where more than human commoning can take place. I therefore see the envelope as a filter of matter, energy, and program. This work addresses firstly the theme of urban water management and the incapacity of centralized rainwater managing systems to deal with climate change-induced extreme weather phenomena. This causes detrimental effects to human society through massive floods that in Europe we're getting more and more familiar with, but also devastating results for urban biodiversity through flash flooding, water contamination, phenomena that are often followed by long-lasting drought seasons. The second theme is loss of urban biodiversity through contamination of resources that are vital for survival, but also through habitat fragmentation, degradation, and the extremely disruptive nature of building architecture that makes it very difficult for species to move, to rest, to forage, to hunt, etc. It is important, of course, to acknowledge the groundbreaking work that already exists at the intersection of those themes, the work of Studio Biocid and Marcos Cruz and Brenda Parker, of course, as well as Richard Beckett with the term probiotic architecture, and the very thorough and intensive work of Studio Animal-Aided Design, and of course the work of many others that are here today. The keynote speech today was extremely amazing in this context. So I study the above from a multiscalar perspective. From my work in landscape architecture, I learned to look at the large scale of cities from a strategy forming perspective. This is the basis of any architecture, a thorough analysis of a site and learning what species inhabit a place, how do they inhabit it, and what do they need, what is missing. In the mesoscale, the scale of the device that I call it, I look into how a specific missing infrastructure can be implemented, but also what is required from humans to change in their daily life to sustain and maintain a design implementation. What scenarios of practicing care can unfold in this intermediate space of the building envelope? And in the macro scale, of course, I look at how we can inform our strategic design interventions from mutualist relationships in nature, but also how can we integrate living organisms in the materiality and the design process so that we can detoxify soil and water and have better conditions for multiple species. This was a design concept for an indoor water and air bioremediation device that would make our interdependence with our own waste, in this case water waste, and other species visible and tangible. I found species that could replace all the different steps that take place in conventional water treatment. But in order to proceed to really designing such systems, I started looking into biology and specifically sea sponges as models for water management and mutualist relationships. This is a sponge from Aliveness, which is an island in Greece with a long tradition of sponge trade. And it's the island where my mom grew up in also. And this is the same sponge from a CT scan perspective. And filtering a bit with biomimetics, sea sponges perform a function called filter feeding. So they have two canal systems, one for incoming water and one for outgoing water. And they have the choanocyte chambers that have some cells that have some tails. So those cells in the chambers move their tails and the water gets in. The water carries nutrients and other organisms. The cells metabolize the nutrients and the other organisms attach on the sponge, forming a coalition of nutrient exchange and grooming. The sponge therefore exposes its whole body to its surrounding to feed. And this, of course, is a process that can lead to a holobiome, so a harmonious, self-sustaining symbiotic system that thrives, but can also be dangerous for the sponge when environmental conditions are thrown off balance. And this is biodesign or bioreceptive design as well, acknowledging the danger and finding ways to mitigate it by altering also our human habits and expectations. Another great source of information for my work is the act of conscious observing as an everyday practice. What can we learn from bioreceptivity from our everyday surroundings? How does the defect on built structures interact with water? And how can this lead to, with anemochory, to biocolonization? Those examples are everywhere. Around us, and they require attunement to them. What can we learn from similar defects in environments of agricultural activity that rely on water? How do different organisms inhabit those spaces, and how do they interact with each other? How can we resist the urge to fix them as well? But also, what can we learn from geological formations that guide the cycle of water, that carries and deposits nutrients for species to grow, even in harsh conditions of solar radiation and drought. With all this being said, I decided to start with a craft approach to translating this knowledge that comes from observing, but also empirical and embodied knowledge about how water behaves on structures through the act of making with clay. I started making small tiles that harvest water, circulated on the surface, and also have a porous texture that allows for water to remain on the surface through also capillary pressure and Laplace pressure in the interfaces, but also for organisms to have a scaffolding that they can grow upon. This hand-building approach doesn't exclude digital design and fabrication, but rather serves as a method of asking questions, revealing challenges, and feeding the design method. In that way, digital and analog are in a constant dialogue and enrich each other. So I am slowly scaling up within this dialogue. This is a larger piece handmade from construction clay that's currently at Humboldt Forum in an exhibition about water. And this dialogue is always between nature, hand building, and digital design. However, water, as I mentioned, is used as a resource for biological growth. And at this stage of my research, the biological growth of lichen biofilms. So in order to receive guidance on the biofilm formation aspect, I started looking into the shift between the terms biodeterioration, and bioreceptivity. And researchers who study the deteriorating effects of microbial biofilms on construction materials, especially in the context of monument preservation, can also be experts on bioreceptivity. So going back to my concept for this indoor bioremediation device, I looked out into lichens as a starting living formation that can trigger beneficial chain reactions for the benefit of all species. Lichens are, as mentioned earlier, symbiotic biofilms of algae and fungi that are resilient, can be found in abundance in nature, fix carbon, and produce very efficiently oxygen. They can also, bioremediate water, and when thought in synergy with a construction material, can form a bioprotective layer. So I reached out to the Materials and the Environment group of the Federal Institution of Material Research and Testing in Berlin, and specifically Professor Anja Gorbushina and her amazing team that has extensively studied biodeterioration, but is also already shifting the perspective to bioreceptivity. We set up an experiment that is currently running to see if we can prove the correlation between growth rates of lichens and the providing of water and specific climatic conditions for the organisms. 1 minute? 0. Okay. I'm closing. Yeah. This experiment does not provide precise repeatability-based data, as the replicates are handmade, but as in landscape architecture, we can't control exactly how public spaces are inhabited, but we can have a very good basis to understand tendencies of growth. And I'm closing with just saying that this experiment is non-exenic. It's not done under sterilities. The small tiles are exposed outside and will be monitored for their photosynthetic activity during the next month. And we look forward to seeing what happens in the future. So thank you.
Thank you. Just please stay on the stage. And I'd also like to ask Nathaniel and Alexandra to go on stage. Thank you. Very good. Let's change. Yes, I hope you have sort of remember we had these 3 different presentations. The last one was more about the water and retaining water and also creating sort of facade elements. Then we had Nathaniel who spoke about the stone and the lichens and the weathering, and Alexandra so presenting her work on why can't I remember now? On the moss, exactly, how, observations on the moss. So are there any questions? Yes.
I have a question for Alexandra. What species of moss do you use, and do you look at the genetic diversity or how they relate to the specific strains that would live in the area where the panels or something would be used? Yeah.
Thank you. Thanks for the question. Yes, we generally tend to choose species that are epilithic, which means that they live on rocks. So some of the mosses that I've been working with, you can also find in the Antarctic, for example, because they tend to also travel. But yes, so they're also called calcifuge in general because they kind of withstand higher levels of calcium. So they are species that you normally already find on building materials, so on bricks, on concrete, and so on. And we choose those because we know that those are more adapted to the more harsh environmental conditions that are at the surface of building materials. It's essentially kind of like a desert environment out there. So we tend to choose those. And those species, there's many of them. I mean, Bryum capillare, Syntrichia ruralis. There's many. But yeah, that's the ones we tend to choose are not the forest ones that sometimes you might see in these kind of more decorative panels, interior ones.
Okay, thank you. Is there any other questions? I have a quick follow-up. What is the color gradient of the mosses you have, not particularly you have studied, but you know of? Because
The color gradient in terms of their color?
Yes.
Yes, so what color is a really interesting one because they change color. So especially species that are adapted to kind of fluctuating light intensity so that they grow in more exposed areas rather than under the forest canopy. So they tend to create certain pigments depending on season in order to protect themselves from high light intensity and ultraviolet light. So what you would see as a very green moss during the winter, you might actually see as something really purple or even gray or black during the summer, but it's the same actual moss. And they do that by creating these pigments and also realigning their chloroplasts strangely in order to deflect light.
Okay, thank you. Yeah, another. Okay, good.
But when the sample of moss that you originally use, it's moss that's adapted to grow on like stone environments, but is like within the specific species, is it from like somewhere in London? Does it live on the roofs? Like in ecology, there's within the species, a specific strain is adapted to that area, like a specific brownfield area of London. Do you take from a specific area in London, or like where does it come from?
So at the moment, the samples that I take, they come from different places, but there's no, because we're trying to figure out the basics of how they kind of photosynthesize, it's not as important for them to be from a particular place, but we do take note of the environmental conditions we take them from, so we know what they're kind of acclimated to. So what you mentioned about strains, maybe it's not necessarily to do with strains, but actually with acclimation, which is different to adaptation. Adaptation means you have another strain or another species, or another strain of the species, but actually acclimation is the capacity of the same species to just change their behavior a little bit according to their environment, which is very different perhaps to other plants. But you might also see this as elongation, like etiolation, so the grows taller in darker environments and so on. So yeah, but genetic stuff is not really needed at the moment for the thing we're doing. It's a bit too niche, and it doesn't really have a reason to, yeah.
Thank you. Are there any other questions?
I have one question, and I'm not fully clear how to articulate it. I was wondering, since you all worked on facades, and recalling the introduction of yesterday's by Petra Gruber for the entire symposium, I was wondering, with regard to time scales and spatial scales, to what extent your research contributes to an understanding of architecture that for sure does not center around the question of the time scales that we humans perceive, but rather at the same time look into very fast life cycles of small living species, and at the same time on geological time, for which buildings usually we do not think of them as geological, but in your case there seems to be a kind of, let's say, strange relationship between these two time and spatial scales.
Who would like to answer?
I'm happy to start. If do you want to?
Yeah.
That's a question for you,
Simultaneously writing a note. It was great, and great question. I liked the framing of building as in conjunction with geological timescales. And I think what's interesting for our project, because it's centered around stone, and humans have this comfortability of permanence, that we were kind of reckoning with that it's kind of countercultural to our immediacy that we crave, or that was joked about within the US, and how fast we want to produce these things from the earlier keynote today. So I think that was something that we really had to reckon with, especially in regards to, say, lichen, we kind of like, glorify or we use as an example for a lot of things, between like air purification and how color is an indicator for it. But these are such slow growing organisms that maybe the benefit of synthetic biology, where we kind of picked up the research, was that we could kind of carry this into a different time frame. That's still quite a nascent field for synthetic lichen, but in concerning of the materials itself, our project wanted to look at a system that was self-sustaining, that could require low maintenance and beyond our typical time scale. Kind of mimicking and reckoning what's already happening within nature, but how can you augment or promote it within a different urban context for a new approach to these things. I guess that's a bit on our project, at least. Do you have anything?
For me, I don't think of time scales in terms of different organisms specifically, because what I'm interested in is always this transformation. So time as a transformative medium, let's say. Yeah, we were talking yesterday also about the mycelium, for instance. It's like when you work with living organisms and lichens, they're really hard to see at the beginning. They're invisible, the biofilms, until they really form. For us as designers, as my friend Emma Zicher says, it's like you yell, "Did you like the food?" And 10 days later, or a month later, or 6 months later, you get a response like, "Yeah, I liked it," or "No, I didn't like it." So that's challenging for a designer. In this fast-paced, we want results now environment or processes that we have built somehow. But I think, as you mentioned, if we want really self-sustaining systems that can adapt to their environment, and this is what is interesting for me, we have to learn to embrace not controlling this time and really tuning into the temporality of the organisms. And yeah, I think that's what I could say.
I have one more thought. I think something that's unique about maybe all three of our organisms that we're using with is the fact that they're pioneering species, and so they're typically the ones that are found in these like ridiculously harsh environments that start that transformation cycle of the nutrients itself. And so through these metabolic pathways, you're then, if we're speaking of working with higher species plants like my collaborator Maria, but I'm over here working with lichen, I know that those two things, they are almost operating in a different time scale. And so it's like the 10-year plan for the project almost. And maybe that's a nice reframing of how you have to plan. It's not necessarily when the building is first erected, but then what is the life cycle of the building itself and its livability, not only for humans. Yeah.
Should I? Yeah, I have a quick, because I'm not sure. Maybe it's going to answer it. Maybe not in terms of the geological timeframes. I think what's really interesting, and we don't usually think about it, considering that mosses are kind of the dinosaurs of plants, and they have been surviving a lot more different conditions than we can even bear to imagine. They will be here long after we're gone, if we do disappear. So I'm quite confident that in terms of the geological time frame, we can't really affect their development. But then in terms of the smaller time scales, there's day-night cycles that obviously influence photosynthetic activity, and that also relates to water cycles and so on within daytime cycles. And then also we talked about the seasonality and the changes that occur during seasons. So we have to have these separate time frames to understand how the organism kind of behaves. But at a geological time frame, we can't really predict what's going to happen because it depends on so many other parameters that we are even influencing sometimes. So, yeah.
Thank you. I think that was extensive. Is there one last question? I would be interested because to all of these presentations, there's a special aesthetic component which is sort of, coming through. And I just wonder if you could give just a quick, each of you, answer about what's sort of the aesthetic trigger, or what triggers you with your research aesthetically most. Just one short answer, please.
Yeah, I think our group kind of reckoned both from the philosophy around this and ethics around our topic, and almost this emotional response that we're quite romanticized with ruin, and we find there's a clear divide of when we invite this overgrowth of nature, or this weathering, this allowance for nature to have its own agency, and yet we don't live that out in our everyday. And so our approach to this particular, I guess, aesthetic was kind of reckoning with, are we okay with witnessing decay, and are we okay without the pristine? And this is ingrained into our everyday living. Like, we go out into the atrium, and it's very sterile compared to some of the images that we've shown. So I think it becomes a really interesting question about what humans are comfortable with and living around. And maybe this slow introduction within the built environment of reintroducing nature within the common urban spaces is stepping stones towards being more comfortable at large with seeing biomaterials that are in a more funky stage of life.
In regards to, so I will interpret this question in regards to the maked thing that I'm making, not in terms of because this was an excellent answer, in terms of the decay or the weathering organisms or the weathering aesthetic. Yeah, for me, I think it was mentioned in the keynote speech, form follows function, but function follows form as well. So I have all those references that I observe throughout my whole life, and especially about geologic formations and the friction with water and how water creates those corrosion patterns or this porosity. And then I have the sponges and the porosity of the sponges. And so I think unconsciously I implement this as a morphological strategy, but function follows form is the principle in a way. And then I get surprised that sometimes I take those principles, I apply them into a material, and then I see new formations in nature that also resemble them. So I think all those things are sort of connected in a way, all those different scales of morphologies from nature. Yeah.
Thank you. It's a great question. For me, I think, to put it in the shortest amount of words possible, I think aesthetics, and more particularly beauty, are really, really, really important for people to kind of take the step towards understanding something and towards valuing it more. So the work that I do in microscopy, for example, is a lot, it's fascinating to me as well. It's really an emotional connection that I feel with the organism when I take these photos. But I also, whenever I show people these photos, they go like, this is moss? And I think I feel that that's the role of the work in a way as well, because if we want to kind of counteract this idea that, oh, we need to scrape mosses off the buildings, we can't do it by just saying, oh, but it's good for this and it's good for that. And no, because people, until they see it and until they understand the beauty and the value in it, and far beyond the instrumentalist reasons, they won't really care, I don't think. We need beauty to care about things, I think.
That was a good final words for this to close this session. Thank you very much. Thank you to all the panelists. Thank you to the people here in the audience. I think we'll make a 10-minute break. Is that okay? Perfect. Okay. In 10 minutes, back here for the final session. Thank you very much.
Okay, so I think we can start. So good afternoon, everyone, and welcome to the session 6, Critical Reflection. I am Gonzalo Vaíllo, affiliated from the University of Innsbruck, and I'm honored to moderate this session. So also thanks to the organization for the invitation to that task. And as its title suggests, this session invites us to pause a little bit and examine the deeper assumptions that underline our contemporary approaches to architecture, and how these new terms, issues, and mindsets that we have been discussing over the past 2 days, like question of circularity, aliveness, what we understand as matter, how they can reflect, how do we reflect and think about those terms in non-conventional ways. So particularly for this session, I'd like to emphasize that this sense of critical reflection is not only a theoretical detour, but actually, I will argue, is necessarily a step for understanding how our practices, concepts, and material transformations and material engagements transform themselves in response to the current issues that we are facing, ecological, technological, and societal shifts. So in this sense, to put it in another way, critical reflections is not only what we design, but how our concepts, methods, and materials might reinforce and shapes these common efforts towards new futures of ecological and social transformations. So each of the presentations that we have 5 instead of 4 like other sessions, each presentation will open a window onto these blind spots that I will argue that conventional approaches, architectural approaches, we might deal with, and propose alternative modes of thinking and acting in how architecture more closely can process terms like adaptations, symbiosis, regeneration, maintenance, multi-species, etc., right? So, as with the previous with the previous panel, the first presenter is not there, but as Barbara said, nonetheless, it deserves an applause as well, at least to be not on the stage, but on the screen. And thank you. So we begin with Stefano Corbo from Delft Institute of Technology, presenting Resilience by Design, which is a pedagogical framework developing at that university to embed adaptability, reuse, and disassembly within architectural education. And then along the presentations run on, I will be presenting the other presenters. So I leave you with the video.
Good afternoon, everyone. I'm sorry for not being physically present with you today, but I very much appreciate the opportunity that you gave me to present my work online. So what I will present in the coming slides is a series of research by design projects I have implemented with colleagues and with students at Delft University of Technology over the past 3 years. And all of the projects that you will see are somehow connected to the attempt to question the role of architecture in confronting the environmental crisis, and more specifically, sea level rise in the Netherlands. So what you will see today is very specific to the context of the Netherlands. This is why I would like to start with one of the many historical reproductions of catastrophic events that have affected the history of the Netherlands and the everyday life of its citizens. Today, although sea level rise is still a constant threat, we cannot talk about catastrophic events anymore. Still, Dutch cities are periodically affected by floods or even heavy precipitations, which are directly connected to climate change, global warming, and also to sea level rise. So over the past years, many have been the initiatives, the government policies, the actions taken in place to question and to critically address the climatic crisis within the context of the Netherlands. According to the Netherlands Environmental Assessment Agency, 55% of the Dutch land surface is at risk of flooding. And if we only talk about sea level rise, that has increased to 3 millimeters per year over the past decades, and increased by 50% compared with the average rate of sea level rise over the 20th century. Even more extreme projections have been implemented to really highlight and emphasize the risk that the entire territory of the Netherlands can be subject to in the coming years, especially when it comes to sea level rise. At the same time, at the university level, at the level of academia, new master programs have been founded to deal with cities, water and climate change, climate adaptation governance, coastal hazards, risks, and climate change and adaptation. But most of these initiatives, most of these academic programs focus on the scale of urban planning, landscape or ecology, but very rarely climate change is addressed at the scale of architecture. And this is what I will try to, let's say, show you in the coming slides. I also want to mention that within the framework of initiatives related to climate change, many municipalities, many cities have started creating some sort of database of interactive maps that can illustrate areas, specific areas of their cities where the risk of flooding is evident and can even cause very specific damage or risks to the urban fabric and to obviously to the communities living in that urban fabric. This is, for example, the Amsterdam Climate Adaptation Map, which very clearly illustrates the risk deriving from heat, flooding, drought, but also, let's say, heavy precipitations more in general. So within this framework, the group of public building at the Faculty of Architecture of TU Delft has ideated 3 years ago a teaching, research and pedagogic model called Resilience by Design. We work with students on architectural design studios. We work on research projects and grant applications. We always try to reflect on what kind of pedagogic tools we can implement to raise awareness on climate change. And we didn't only teach and write, but we also created some sort of online databases. We worked on exhibitions. We worked on apps and multimedia forms of dissemination. The framework of Resilience by Design are the 17 UN Sustainable Development Goals, in which somehow sustainability, or let's say sustainable cities and communities, are one of the main pillars, or let's say the main points of the whole UN agenda. More specifically, we somehow borrowed the word and let's say the connotation that resilience has for the future of the built environment from one of the UN Habitat World Cities report, in which resilience is conceived and presented as one of the keywords to understand the future of urban environments. As a resilient city is for the UN Habitat program, in fact, one that is able to absorb, adapt and recover from the shocks and stresses that are likely to happen, especially due to climate change. We try to apply this definition to the scale of architecture, to the scale of public buildings, so how public buildings can be designed, built, and how they can adapt to future shocks. This is the premise and the framework of resilience by design. As you can imagine, resilience by design echoes research by design as a way in which design and research are completely entangled, in which theory and practice, analysis and speculation come together to conceive and develop architectural ideas. Resilience by design presents both projection and speculation as a mean to prepare, describe, and explain. So through resilience by design, we attempt to let's say question or to pose the following questions. How can public architecture resist future shocks and be prepared for climate change? How can public architecture adapt to unpredictable scenarios? What special and material characteristics does resilient architecture need to have? For this reason, in our research projects, but also in our, let's say, courses, we work on specific design strategies like adaptability, design for disassembly, modularity, flexibility. And each of these design strategies is including a series of specific techniques of operative solutions, like the use of non-load-bearing partitions, the application of wet-proofing materials, exposed connections, mechanical fasteners, new forms of circulations. So students work on these techniques and on these design strategies and apply them to the design of public buildings, of resilient public buildings. Before jumping into the design of buildings, students obviously implement a series of research activities that have to do with the analysis of existing literature, but also with case studies, which have to do with typological explorations on how we can design new typologies that can embed resilience as one of the main criteria or guidelines, if you want, but also more, let's say, concrete analysis of buildings in which not only composition, form, and expressive elements are analyzed, but also their performance and their environmental behavior, so to speak. And also, students work at the scale of matter and materiality to understand what kind of properties materials can have and can embed in order to create architectures that are really resilient and able to deal with climate change. So the end result, let's say, of these explorations paves the way for a sort of threefold structure, which is what we try to apply specifically in our studios and in our teaching activities. Let's say, our architectural design studios that are connected to resilience by design, students work throughout three main phases. In the first phase, they come up with a prototype, a sort of universal model that serve as a special module or unit illustrating the characteristics of resilient architecture. And in the following phases, they try to transform this prototype, this abstract model, into specific buildings that are responding to site conditions, to context-specific needs, and to demands and necessities from local communities. So the whole studio is based on this kind of dichotomy between the global and the local, the abstract and the site-specific. And these polarities, create a sort of dialogue between, let's say, the principles of resilient architecture and then their applications into real-life scenarios. So these are some of the abstract prototypes that students have developed over the years. In each of these prototypes, students try to reflect on constructive systems, skins, or envelopes, performance, or let's say environmental behavior, internal layout and composition. All of these different layers pave the way for the definition of a resilient prototype. At the same time, as I was mentioning before, these prototypes also deal with specific strategies like flexibility, adaptability, design for disassembly. Here you can see an example of how the prototype can be made of different pieces and elements that are easy to assemble and disassemble. Or, let's say, twofold systems in which part of the prototype is permanent and is ready to be colonized by water and by let's say, non-human forms of life, should sea level rise be extreme in the Netherlands, while a second element or a second component of the prototype is easy to be dismantled and transported elsewhere. Or like in this case, the prototype comes in as a sort of module, as a sort of 6x6 unit that can be, let's say, generating entire buildings out of the same module by extension, by addition, or by subtraction, according to the necessities of the site in which this prototype is implemented. So out of this 6x6 module, a compact building which acts as a continuation of the urban fabric along the water and above the water can be built, but also the same unit can generate a series of pavilion-like structures that act as floating pavilions in the island of Terschelling in the Netherlands. To illustrate you the entire process, I want to, let's say, show you an example of a prototype that dealt with animal-aided design. So this abstract model was conceived to explore how architecture can embed non-human forms of life, plants, insects, animals more in general, that can, let's say, generate and describe a sort of ecology of care or ecology of companionship. So the students started understanding and analyzing the flora and the fauna of the two sites in which she implemented her prototype and tried to understand how that prototype could embed and accommodate not only plant species, but also birds, mammals, reptiles, amphibians, and other non-human forms of life. The end result in moving from an abstract prototype to two different buildings in the city of Amsterdam was, in the first site, a sort of longitudinal building generated out of the same abstract prototype, which was accommodating cultural and art events. And in the second site, the same prototype became a sort of parasitic structure which was built on top of an existing historic building in the city center of Amsterdam, which could increase the biodiversity of the entire area. Students, of course, always try to visualize how resilient architecture can actually resist the threat of climate change and how can embed and deal with water. So this is an example of how the same student working with the prototype on animal-aided design envisioned her own architecture in 2025, and then even in the extreme scenario of 2017, when maybe water will take over and will invade the space of the city. More and more visualizations try to test the adaptability of architecture to climate change and to the, let's say, the threat, but also the opportunity that water can, let's say, offer for our environment and also for architecture. Overall, the students with these resilience by design projects try to learn to design for and with climate change and to hopefully propose an alternative model of architecture that is more inclusive, more sustainable, and also more critical with the historic, cultural, and economic premises that have caused climate change. Thank you so much.
Okay. So now you know how to escape time warnings, is just bringing a video. Now we know next with Bastian Scheile from the Technical University of Aachen, who will take us with the presentation meeting Saltnotes in Elevations, who trace how industrial infrastructures reshape ecological life and cultural imaginaries. So looking forward.
Yes, thank you. Hello, my name is Bastian Schleier. I'm a carpenter, architect, and a research associate at the Academic and Research Department for Artistic Methods at RWTH Aachen. And I have just begun my PhD project, Machine-Smith Metabolisms, in October. So what I'm presenting today is less a report of completed results and more of a vision of my research ahead, grounded in work I began at the Technical University Delft in my master's thesis, where the salmon walks up the staircase. So I'm like in that white gap at the moment. My research is taking place in the Murg Valley in the northern Black Forest, following the river Murg, 80 kilometers from the Rhine upwards its stream towards the mountains around Bayersbronn and Freudenstadt. The valley is characterized by canyon-like sceneries, where the river carves its way between granite cliffs and boulders. Alongside the landscape runs a dense mesh of infrastructures following the stream through the valley. It is this dense mesh of natural and technical life forms, a co-becoming across craft history, industrialization, and ecological restoration that I will investigate in my research. I approach life not merely as a biological phenomenon, but as a media-ecological process. I argue that this perspective can help us rethink the social and ecological challenges facing rural regions today. For today's discussion, the story of the Atlantic salmon will guide us. Until the 18th century, the Murg was one of the most important salmon spawning rivers in the Upper Rhine system. The clarity of the well water coming from the mountaintops, the steepness of the course, in parts unreachable for humans, the granite boulders, all these factors provided an ideal milieu for the salmon's final journey to produce offspring. For romantic thinkers such as Schelling, at the time, landscapes like these became metaphors for a self-organized, unified, harmonic conception of aliveness as a living nature. The universal organism of nature is animated by a common life. Each particular organism is but a repetition of the eternal act of productivity. For the growing urban communities, it was rural areas such as the Black Forest that represented a simple life in harmony with nature. Yet, this was mostly an idealized projection rather than an accurate reflection of life in the valley. In contrast to this romantic vision, the landscape was already deeply shaped by human labor. Timber floating, for example, had been the strongest economic actor in the region for centuries, supplying the rapidly growing cities and their ship fleets with fir and spruce logs from the Black Forest. Like many rural regions, the valley had long been structured by resource extraction and was therefore already a highly infrastructural environment. The forests were systematically logged and replanted as monocultures, resulting in ecological stresses such as severe bark beetle damage that is increasingly visible today in the context of the climate crisis. Nevertheless, rural landscapes continue to be romanticized, and the pre-industrial livelihoods that shaped them are often reduced to folklore, rather than recognized as inherent to urban industrial developments. From 1800 onwards, due to ever-larging urban demand for reliant resource flows, the methods of extraction changed and intensified rapidly. Large media infrastructures emerged throughout the valley, such as paper factories, energy plants, pipeline systems, and hydropower dams, transforming the river more and more into a kinetic machine. It was also during this period that the salmon disappeared from the valley. These technical systems did not only transform human living environments, they also established new ecological rhythms and regimes across the habitats of many non-human species. At the same time, the tacit forms of knowledge embedded in craft practices increasingly disappeared, replaced by an engineering mindset focused on control. In this view, life was reduced to a thermodynamic process, a flow of energy to be optimized and regulated. Or as physiologist Etienne Jules Marais put it, every movement of life is but the translation of a vital force into mechanical work. By now, many of the infrastructures have lost their economic relevance, became obsolete, and many left abandoned. While often being considered problematic heritage, these structures have become an additional techno-geological layer to the landscape. Failed attempts to reinterpret them as tourist distractions point to the question what their role in the future might be. In the 21st century, something remarkable happened. The Atlantic salmon successfully returned to its native spawning grounds after a 200 years absence due to a broad range of reintroduction efforts. This success is embraced throughout the region, and a salmon-themed hiking trail has been established, celebrating the return with messages like "nature is back and healthy again." Yet, this success has only been possible through an interplay of natural and technical actors, improvements in water quality, monitoring systems, and especially fish staircases. While these now allow fish to navigate upstream, they also carry a deeper message. They blur the boundary between natural ecosystems and human-engineered interventions in support of ecological restoration. This is where a new conception of aliveness becomes productive, as philosopher Gilbert Simondon reminds us. The living being and the technical object are both individuations of a more fundamental process, that of becoming within the milieu. This becomes all the more apparent when looking at the Murkai pressure plant, where a new fish elevator has been installed. The plant depends on the river's ecosystem to operate, yet for around 100 years, it recursively shaped that same ecosystem in ways that denied the salmon access to its habitat. This example highlights a crucial insight. Technical and natural forms of life are not separate, they co-evolve within a shared milieu. This fish elevator is part of a vast infrastructural system for energy extraction, yet it now also functions as a mediating apparatus for ecological passage. It is a technical object, but it is simultaneously part of the landscape and the salmon. In this sense, it acts both as a prosthetic organ and as an artificial environment, complicating our definitions of nature, technologies, and bodies. I argue that it is precisely these complications that offer an alternative to the persistent romanticization of rural landscapes. Or as media theorist Jussi Parikka reminds us, media are not outside of nature, they are nature reorganized. This insight can help us reconsider the pre-industrial and industrial not as discrete periods, but as part of a continuous process. Both novel infrastructures and pre-industrial tools and techniques are not mere carriers of media substrates. They are active forms of media themselves. They shape perception and knowledge. They enable or constrain the existence of human and non-humans alike. Timber, wires, dams, fish ladders, salmon, and humans all become part of a single media ecological entanglement. In this shared mode of becoming, hybrid forms of life generate ecological rhythms and perceptual worlds. Life then is not merely a property of individual organisms. It emerges in the relationships between all things. The Murg Valley is thus more than a site of industrial heritage or ecological restoration. It serves as a model for rethinking rural spaces as media ecological beings themselves. Here, media theorist Marie-Louise Angerer can help through a more accurate conception of aliveness. What we call life is not confined to the biological, but is the modulation of intensity in a field of relations. And so my project asks the question, how can we reimagine rural landscapes not merely as extractive operations or places of folklore, but as media ecological archives that point towards a future of coexistence. Thank you.
Thank you, Bastian. Now I would like to introduce Luisa Svetno in Fermenting Architecture, who draws from fermentation to proposessive, from accelerated, control-oriented designs towards slower, relational, and context-specific approaches.
Yes. Also a warm welcome from my side, and thank you for joining me today for my talk about fermenting architecture. This work is initially my diploma project at the TU Vienna. It's a research, a practice, and a way of thinking. It proposes a radical shift where architecture should no longer be object-driven, but process-driven. At its core, the project is about relational thinking, understanding the dynamic relationships between processes, materials, and care. Before I go deeper into this work, a little bit to my background that formed my personal starter culture. My starting point was entirely practical, and food was my entry. Through cooking and fermenting, I learned first about processes, timing, material behavior, and failure. Here, matter teaches. What struck me early on is that people working with food, like farmers or fermenters, have kind of a systemic understanding for the environment because they're acting as part of it. But shouldn't architects have it too? Aside from that, 4 projects also contributed to my personal starter culture. Each one confronted architecture with real constraints: waste, soil, time, and climate. Together, they shifted my perspective from designing objects to working with processes. Beginning this research, I first dived deeper into fermentation, and here Sándor was crucial. He understands fermentation as a worldview: relational, slow, context-specific, and collaborative. This view provided a framework for rethinking architectural practice. The theoretical foundation combines new materialism, ecological practice, and process philosophy, and drawing on authors like Bennett, Ingold, Howe, and Thing, they helped articulate what fermentation already shows in practice. Materials act, environments co-shape outcomes, and knowledge is relational. Better? This means my personal starter culture should be read in a diffractive reading method, characterized by Karen Barrett. Reading it diffractively means letting theory and practice shape one another. Insights emerge through the entanglement, not by separating reflection from action. This leads to my central question: how can the principle of fermentation, understood as both an epistemic and practical model, transform architectural process into a relational, processual, and caring practice? So, what is fermentation? Fermentation, in its most basic sense, is the transformation of food by microorganisms breaking down sugars into new compounds. It's a biochemical process and one of the oldest preserving techniques practiced worldwide. What matters for architecture is not the biochemistry, but it proposes a different logic of making. It's condition-based, non-linear, collaborative, it can't be rushed, and it resists total control. Fermentation shows the transformation happens through interactions. Architecture could learn from this shift. The presentation follows the 5 phases of fermentation: start, mix, rest, rise, and cultivate. They help to rethink architecture as something that unfolds, changes, requires tending, instead of being designed as a finished object, but as a living process. So here, start is the first phase of fermentation. This is where the ingredients, environments, and intentions meet. In architecture, this is the moment where decisions begin to define entire futures. And this work starts with a feeling of discomfort, that the current architectural practice is out of sync with the ecological and social realities. Climate breakdown, extraction, acceleration, these are not eternal issues, but problems rooted in how we build, how fast we build, and the systems we rely on. All of these lead me to the question if there isn't another way of understanding architectural practice. This question, together with my personal starter culture, allowed me to recognize the parallels between building and fermenting. Across all projects, the same patterns appeared. Building and fermenting are both processes transforming raw matter, bringing together people, tools, and stories. They are slow and iterative, depending on materials' behavior, and require adaptation and care. All of this formed the seed to my work. In the second phase, mix, ingredients are needed together, in fermentation and in architecture. This chapter examines how architecture is situated and embodied, and food in its broader sense becomes here the main teacher for this. So what can we learn from food? There's an old saying that architecture is the daughter of agriculture. This reveals an ancient truth. Food is grounded, site-specific, and relational. It forces you to work with soil, season, microbes, and climate, and architecture, in contrast, often abstracts these realities into drawings and regulations detached from its context. Torah beautifully exemplifies this and shows how place imprints itself into matter. Just like no sourdough or wine tastes the same elsewhere, architectural materials also carry geologies and histories. Context should be understood as a living ecology, not as a site boundary. Here, bioregional thinking is what's needed. It reframes regions through soil, climate, vegetation, watersheds, and local waste streams. Architecture should work with these ecologies rather than import generic solutions. Adjusting to every new terroir also means learning from failure and trying.
Fermentation depends on microbes, temperature, time, and gestures, meaning you cannot standardize it. Things fail, conditions vary. Architecture should accept this variability, adjusting design through ongoing feedback rather than forcing predetermined outcomes. Fermentation relies on tacit knowledge, knowing through the hand, through touch, smell, and texture, like a baker who knows that the dough is ready to proceed just by touch. It's the kind of knowledge where we know more than we can tell, as Michael Polanyi said, who introduced the term tacit knowledge. Architecture often dismisses this embodied intelligence, yet building also depends on humidity, density, tensions, all things learned through direct interactions with materials. Cooking, fermenting, and building are collective acts. They create temporary publics, groups of people acting together in solidarity. This collective dimension decentralizes authorship, and agency is distributed between people, materials, and conditions. So in mix, knowledge and place revealed is safe as relational. Now we move into rest, a slower phase where materials start to act on their own, invisible transformation takes place, and where architecture must learn to step back. Slowing down is not passive. It exposes dynamics usually hidden in fast architectural workflows. To pass on tacit knowledge, to act as a collective with open processes, all of this needs time. But as in fermentation, slowness here means richness in flavor, in texture, and in outcomes. Rest allows materials and processes to continue to transformation outside human control, demanding a more responsive practice. Drawing on Jane Bannett and Tim Ingold, materials are vibrant and alive, never inert. They move, shift, absorb, and resist. Recognizing this challenges architecture's obsession with control. Instead of enforcing form, we can work in coexistence with material tendencies, correspond with it, and share agency. This leads to Karen Barrett's concept of interaction. That means materials, tools, people, and environments emerge through their relations. Projects should arise from the entanglement of all actors, human and more than human. After start, mix, rest, we now reach rise, the phase where processes become visible. In fermentation, rise is a response to the conditions we set. In architecture, this means shifting roles, designing conditions, and accepting the outcomes that emerge through processes. This is the moment where the role of the architects shift. Less control, more facilitation. A fermenter doesn't command. They observe, adjust, and respond. They work with uncertainty and allow conditions to shape results. The mindset transforms the architect from author to facilitator and from controller to collaborator. If one can't control the outcome, you can only set the conditions around it. This is what fermentation teaches you. Translating this into architecture means designing frameworks where things can entangle. Local materials, available labor, waste streams, and seasonal rhythms become design parameters. Iteration replaces prediction, prototypes replace renderings. Care is embedded in fermentation. Feeding, checking, adjusting, and architecture is no different. If we understand buildings as ongoing processes, then maintenance becomes design, not an afterthought. Care time replaces the build and abandon mentality that dominates the industry today. At my exhibition, in the collaboration with the association SU, construction and fermentation took place side by side, making the parallel acts of making visible. Humidity, temperature, and the unfinished space set the pace. The site became a co-producer, not a backdrop. With rise, the ferment becomes active. Now, we move to cultivate, the phase where processes are sustained, fed, and carried forward. This is where architectural practice becomes long-term, iterative, and collaborative. If architecture behaves like a ferment, it never fully ends. It circulates, adapts, ages, and transforms, and practice grows through repetition and feedback, more like a sourdough starter than a fixed plan. Fermentation spreads through sharing, passing starters, exchanging methods, and learning collectively. Architecture can function in the same way, through open processes and shared infrastructures. Cultivating means leaving space for future hands, future adjustments, and future conditions. Fermentative practice grows like a starter culture with care and shared responsibility, where materials become a beginning, not an end. Time becomes an ally rather than an enemy, and cooperation replaces control. So, what do we need to ferment architecture and cultivate relational, processual, and caring practice? We need to slow down, we need to practice with care, act as a collective, design conditions, not objects, use the existing and value the process. Architecture here becomes a metabolism of care, slowness, and shared knowledge. And like any ferment, this work does not end here, it continues to evolve. Thank you.
Thank you, Luisa. Then I would like to welcome Paul Trecht from the Department for Planetary Education with a paper designing for deep time around the question of multi-species maintenance.
All right. Does that work? Yeah, all right. Architectural longevity has traditionally been framed through ideas of resistance, the building as a fortress, structure as shelter from time, weather, and decay. This conception, inherited from modernist and capitalist paradigms, continues to inform even progressive design. Yet architecture's material existence and ecological realities resist this narrative. No building escapes maintenance, and no building is maintained by humans alone. We already live in multi-species cities, just not the ones that we imagined. Today, I propose multi-species maintenance as a core framework for architectural and biodesign practices. Rather than longevity as resistance to change, I argue for regenerative longevity, where architectural survival depends on an entangled ecology of care, decomposition, and adaptation. This means challenging ownership, purity, efficiency, and the assumptions that buildings can or should remain stable. It means understanding architecture as a process, not as an object. Contemporary building practices treat maintenance as a technical afterthought, a nuisance. Longevity is framed as structural virtue. Buildings are designed to withstand time, moisture, wear, rot, and the temporal messiness of living systems, to eliminate the presence of the living. In this outdated framework, the spatiotemporal is an enemy to be conquered. Maintenance becomes a necessary evil, repetitive, low status, external to design. Their maintenance and contributions are supplementary rather than constitutive, and innovation is fetishized. Even in sustainable architecture and biointegrated systems, longevity is still defined as optimizing resistance. Life is harnessed under controlled conditions. Green facades are monitored, bioreactive panels are calibrated for performance only. These projects remain tethered to the logic of control, simulating natural systems while keeping their unruliness at bay. The underlying model of architectural value hasn't shifted. Buildings are expected to resist nature even when they incorporate it, and when buildings are designed to resist change, they become extractive. But what if maintenance is not secondary but foundational? It makes infrastructures visible, not as systems of pipes and cables, but as lived relationships. It's intimate, situated, and slow. And crucially, it's not exclusively human. Fungi maintain soil health through signaling and nutrient exchange. Mosses modulate humidity on the surfaces they grow on. They're not metaphors, but they are maintenance practices that sustain life. A building is therefore a temporally extended assemblage, constantly cleaned, repaired, adjusted, fed, inhabited, and transformed by human and non-human actors. Maintenance becomes how buildings participate in life rather than shield against it. And even when materials have shorter lifespans, from compostable bricks to mycelial panels, maintenance doesn't disappear, it intensifies. Because the materials live and die faster, they demand more responsive forms of care across scales and species. This makes maintenance a hyper-object, vast, decentralized, and impossible to grasp fully, operating across scales from daily cleaning to structural decay over generations. Erecting a building is a momentary act. Inhabitation and decomposition unfold over deep time. And like all hyper-objects, it implicates us. It exceeds human perception, and we are embedded within it as agents, obstacles, and beneficiaries. But maintenance resists simplification. It's messy, uncertain, and political. And as Hélène Frichot reminds us, this messiness should not be eliminated but embraced. Buildings are always in the making, always in decomposition, always in negotiation. Consequently, maintenance becomes an ontological process. As an ontological process, its presence is inseparable from the flows of energy that sustain it. Every act of maintenance is a transformation of attention, time, and energy, representing exchanges within the metabolism of our built environment. Yet they remain invisible, unaccounted for in architectural narratives and sustainability metrics. We measure buildings by their embodied carbon, but ignore the energy flows that keep them alive. Recently, however, these energy flows started to suffer from sludge, to use behavioral economic terms. Deliberate friction built into systems to slow, discourage, complicate, and offload the necessary processes, usually for capital gains. In maintenance, this physicalizes through proprietary tools, obscure warranty systems, opaque subcontracting chains, and deferred budgets. I term this maintenance sludge. What could be an act of empowerment becomes an obstacle course, draining the social and psychological energy. No system maintains itself. Every object building a network is entangled in another's energy. Designing for multi-species maintenance then shifts attention from users to maintainers and from systems to compatibility and entanglement. And materials affordances are emergent, relational, and situated. A surface that hosts microbial life in one humidity regime may repel it in another. Behavior changes with context. And this means rethinking the building layers away from static steel components and towards assemblages. Maintenance becomes how we interface with the shifting ecologies, where oysters monitor water quality, microbes regulate humidity, and humans are just one actor among many. It also requires new tools and methods, understanding decomposition as a regenerative phase of design, and acknowledging that black boxing and patenting building elements is an ethical failure because it prevents access to maintenance knowledge and participation. So designing through multi-species maintenance subsequently invites a new aesthetic, labor, and material language, creating a palimpsest vernacular, architecture that draws not from its finished form, but from layered adaptation, where traces of care, decay, and repair are visible, and speculative maintenance is legible, a collaborative interspecies architecture, one that rejects seamlessness and favors legibility of process. Materials are chosen for their ability to signal or participate in transformation, to discolor, decompose, and host other life. This challenges existing norms where functions remain rigidly separated, in contrast with the entanglements found in natural systems. In current practices, the human maintains a separated layer, or a specialized human maintains the fused layer. Broken layers are exchanged, and any benefits from composting, for example, happen elsewhere. In my proposed practice, the organisms maintain themselves and the surrounding layers in cycle, not a linear, but a generative-degenerative process. I termed these configurations entangled building assemblages, serving as conduits of energy and matter linking materials, organisms, and environments. Rainwater seeps into green roofs, carrying nutrients, including those deposited by urban wildlife, such as pigeons. Within a wall, the slow decay of one layer releases minerals and organic compounds, feeding the network of another layer, let's say a living mycelium insulation. These, in turn, redistribute nutrients and stabilizes the wall's microclimate by strengthening moisture-regulating biofilms, acting as responsive vapor-permeable layers, absorbing excess moisture and releasing it in dry periods. These systems invert the logic of the passive membrane. Instead of sealing out the environment, they are in dialogue with it. The multi-species actors participate intermittently. A gardener adds composts to supplement deficiencies, or sensor-guided micro-irrigation, or microbial colonies responding to shifts in temperature and light. Energy flows through these assemblages as chemical, thermal, and biological transformations, circulating between materials, organisms, and humans. These metabolisms occur on different scales and temporalities, and while some of these examples that I just gave are more achievable than others in the now, it visualizes ways in which our structures can be situated and respond to their context. The vernacular emerges from an ethics of responsibility, the ability to respond and remain responsive over time. But this means diversifying the microbes, fungi, plants, and humans that we work with, choosing hyperlocal and becoming deeply ingrained in the social fabric and labor practices. And this is not in romanticizing the informal or the ruin. It is cultivating an architecture of entanglement where regenerative longevity emerges from embedding change that also affects the system of ownership. Under capitalist regimes, buildings are commodities and maintenance devalues them. But maintenance is what makes value possible. To embrace multi-species maintenance, ownership must give way to stewardship. Buildings are nodes in complex socio-ecological networks, and maintenance becomes the space of society, holding conflict, friction, and different temporalities. And as Puy de la Bella Casa writes, we care for things not because they produce value, but because they already have value. So to embrace multi-species maintenance, we must move beyond the owner-user binary. Ownership implies control and the desire to minimize obligations. We, however, must stay with uncertainty, to have negative capabilities, as the poet John Keats calls it. Despite my proposal to remain with uncertainty, though, I do want to offer a look into the now and the future. The now means recognizing that every decision we make enters a temporal lag. The average building takes years from conception to completion, and by the time it's done, the context has already shifted. So designing today requires anticipating transformation, not resisting it. The future is not a fantasy of self-healing, self-growing autonomous buildings. That's another delusion of longevity as resistance. A world that maintains itself is also one that no longer needs us, and a world without relation. So lastly, the question is not whether buildings require maintenance, but what kind of maintenance we choose to enable. In the context of this symposium, that means that whatever strategy we propose, without planning for its maintenance, it's a strategy prone to fail. Buildings are never finished. They are not singular events, but ongoing processes, routines, decompositions, adjustments, surprises. Their lives are defined not by how long they can resist change, but how well they participate in it. Regenerative longevity is not independence, but interdependence, not control, but care. And multi-species maintenance is not a limitation. It's architecture's most radical possibility. Thank you.
Thank you, Paul. And then we go now for the last presentation by Timothy Ryan, who will present Artificial Dark Earth. It's a way of repositioning nature, I would say, not as a resource, but as a hybrid, semi-domesticated entity. So very much looking forward how you unfold that. Thank you.
So yeah, good afternoon. Thank you for the invitation to speak on some of the work done at Leeds. Pardon me. So yeah, I'll try to compress about 10 years of research in 10 minutes, so wish me luck. So basically centered around the questions of carbon as a building material, soil as a spatial condition, ecology as site and vice versa, and microorganism as resident. So we begin in Amazonia, where there exists a huge diversity of indigenous cultures with sophisticated forms of ecologically integrated urbanities. This is just a small part of Amazonia, and still not so small at all. Each tiny dot here representing an entire square kilometer of rainforest, and amongst it, 3 protected indigenous territories become visible, emerging as antibodies against the viral encroachment of linear resource extraction. There are many reasons for this, but one of the keys is in their soil and the production and management of this uniquely dark, rich, fertile soil known as Amazonian dark earth. It's a powerful example of cultivating matter. It's a result of many things, amongst it shifting cultivation, some semi-domesticated agroforestry, and the reworking of animal, vegetal waste back into their soil. Amongst it, though, at the very heart of it all is the use of pyrolysis, which is an anaerobic form of fire, and using oxygen starvation transforms organic carbon down at the molecular scale. Carbon fuses in these conditions into aromatic rings, forming something called black carbon, which is durable and recalcitrant, meaning it's resistant to decay. Archaeological finds evidence to this and show its persistence in soil for millennia. Under these conditions, it expands and solidifies into an incredibly complex structure. A single gram of it, about this big, can have thousands of meters of surface area within it. These intricate networks perform absolute wonders for the soil, augmenting water retention, increasing cation exchange capacity, and improving soil nutrient adsorption within the soil. And this forms an optimum environment for bacteria, fungi, and other microorganisms to flourish within these structures, dramatically increasing soil fertility, soil diversity, and therefore kind of the vitality of the biome above it. Through pyrolysis, indigenous cultures were allowed to bolster their ecological productivity, ensuring food security and the growth of their civilizations. So when colonialists arrived to the Amazon, they did not find Amazonia as a hinterland of nature untouched. It was already home to over 10 million inhabitants. The primary effort conducted at Lees is to honor this mode of existence and to translate the properties of Amazonian dark earth into artificial dark earth, into kind of carbon-based architectures. This builds upon a rich legacy of earthen architectures by imbuing black carbon into soils and clays as a novel hybrid of ancient materials. These structures not only promise kind of a scalable form of carbon sequestration, but they're also able to directly engage with the elements, being able to digest atmospheric pollution or filter contaminants out of water, and perhaps most interestingly, able to fertilize the soil from which they are built. Lees is kind of this process and an ongoing study of an immense amount of different of these hybrids of materials. One of them, for example, here demonstrating that using pyrolyzed oak and beech wood from forest waste at a 1-to-1 mass ratio, and this is using maybe about 80% purity of black carbon, signifying that a single kilogram of this material represents 1.4 to 1.5 kilograms of CO2 absorbed from the atmosphere, which is completely different than concrete, which may release a kilogram of CO2 for every kilogram of concrete made. But beyond its sequestrative capabilities, it also transforms this dense network of clay into a greater network of micro and macro porosity throughout the material. Through X-ray analysis, a single kind of sliver, so a micron width, demonstrates the presence and depth of carbon throughout the material. Each one of these pieces containing an entire universe of networks happening in all directions. A three-dimensional X-ray begins to demonstrate these universes working together. So this really transforms the idea of architectural material into architectural medium. This intention can be tailored as gradient materiality from kind of dense earth at the bottom to a lighter porous structure at the top, as well as across the thickness of a wall, questioning the boundary between interior architecture and outer ecology. So, you know, strong, safe, warm where it needs to be, and biodegradable, wet, teeming with life where it needs to be. So we test kind of lots of different hybrids of these materials, some from forest waste, in this case of Scott pine, with its barks, needles, cones, etc., as well as the mutualistic bacteria, yeast, etc., that come with it. It can also be coming from you know, wild ecologies or urban streams, as well as agricultural cases. What becomes interesting too is as these clays become fired in ceramics, all their organic principles, heightening tensile or compressive strength, can also be used in their pyrolytic form, creating pockets or conduits of carbonaceous networks, as well as transforming certain forms of carbon, like in calcium carbonate, into grogs and fluxes, which interact with the chemistry of the material. And there's an evident connection between pyrolysis and ceramics, this idea of fire, converting either green carbon into black carbon or clay into ceramic. And it's an eternal kind of tango, a material tango happening at the biological and at the geological scales. And it's also delightfully complicating this notion of boundary between the inside and the outside, turning architectural surface into architectural substrate, so being able to actually go within. So here you see kind of the inclusion of carbon within the dense network of clay. And then further, these can be inoculated with microorganisms, so using nutritional medium and microbial cultures, placing material into bioreactors, and saturating them with moisture, with nutrients, with microbes, and filling their pockets as reservoirs for microorganisms to be able to emerge. And these lay the foundations for entire ecological networks, starting from the very little, and then from it are able to emerge the mosses, the lichens, liverworts, and later on ferns, plants, etc. How does this kind of material then inform a greater architecture of dark earth? Because it must deal with these notions of ecology, but also the age-old questions of shelter, so about gravity, about structure, and human occupation. So these are able to invite ecology, but also invite human occupation in a new way. So they must be still load-bearing, but possibly also loan-building, and helping us grow food or to develop health within our architectural environments. So questions of porosity and surface augmentation, humidity control, woven into these ideas of structural integrity. It seems that the most appropriate way is to have the material actually dictate how we choose our design parameters. So this was a prototypical wall panel where carbon is identified within the material and dictating tool paths for a 6-axis robotic arm to carve into the material and generate channels from which water and nutrients can be directed towards the carbon where it works best. And this can then therefore inform entire walls, buildings, perhaps even at the urban scale, of spaces that are respiratory and remediative. And this can be spatial too, where columns and foundations become reservoirs for water and for nutrients from microorganisms to exist and for greater ecologies to emerge. So a big kind of challenge here at least is to, and you'll see some of the smaller pieces, but they're now getting quite large, is to create a kiln that both pyrolyzes organic matter but also fires these carbonaceous ceramics. And inspired by the mechanics of pyrolysis, to create different size kilns, different shape kilns, dual retort kilns, open flame kilns with their own requirements for structure, but also becoming walls and columns, having joinery, having components, all this architectural language, and slowly becoming a starting point for a new form of pyrolytic architecture from which a kiln dictates the entire functioning of the creation of a building. So starting with the principles of earthen architecture, which are kind of heavy and massive at the bottom, but slowly being instructed by their instrument of fire, as a nod to Gottfried Semper, where fire becomes the origin for architecture. This is a temple, a consecration of carbon, a kiln that is now inhabitable, a space of study for the preparation, for the processing and fabrication, and for the pyrolysis of carbon, an interplay between the inert and the living, dark earth architecture as soil, as a living being, and as a place for us to exist in a new way amongst our ecology. So thank you for your time.
And then I like first to ask the audience if they have any questions.
Thank you. Wonderful presentations. Thank you so much. It's so inspiring to see the angles that you all take. I think my question is to do, because a lot of you have been speaking about the vernacular in some form or another, and kind of practices that kind of have their roots far into the past, but some are still today. We still have them. I guess my question is, how do you relate to the rural environment? And some of you already spoke about this slightly, but I guess my question is, if we know that these techniques and the vernacular ways of making are so important and we need to bring them in cities, How important is it also to preserve them in the rural environment? And how important is the rural environment for architecture in general? Because we don't see much of that actually being tackled by contemporary architecture.
Then I try to answer your question. I think the rural architecture shows us some ways of how we use what exists. And this is what we try to apply maybe in cities as well, but it doesn't mean that we have to ignore it or forget it in the rural areas. So vernacular architecture, this could be something like, we need maybe some 2.0 vernacular architecture where we go back and learn from all the techniques, histories that has been built up. So we can actually then also apply what we learned in the meantime and combine all of those things. So, because I think just by applying or bringing together all of the things we know by now, this makes sense. We can't overgo it. There's no binary between the new and the old. It's just a way to bring it together. So if you go into the rural area, you build with the things you find there. If you work in the urban area, there are different things you find. So just need to walk with what's there. Maybe you talk about it.
I mean, yeah, so I see it quite similar. I think that, very similar to a lot of ecosystems, right, that are destroyed. There's a lot of vernacular that's destroyed, that's not going to come back. So we kind of need to find new vernaculars that fit for the future and the way we're operating in right now, whatever that means. It can be with living organisms, it can be with whatever, but we need to find new vernaculars for that. And at least in my instance, I kind of chose the built environment as cities, because that's where most of the human, non-human interactions happen. But obviously the rurality is incredibly important in that. But it's it's a slightly different way of how things are being used. But I think especially I feel both from a fermenting perspective, but also from a maintenance perspective, the rural deals with maintenance a lot different. In the city, it's more very capital driven, where we just want it quick and we don't want to do anything for it. Whereas often in the rural environment, we understand that we need to regularly tend to things, like you were saying about agriculture, right? That's not something you just do and then you're okay, cool. But you have to tend to it, you have to regularly maintain things. And that goes for structures as well. And the structures, if you look at a farm, it's not one building designed from start to finish. It starts with a little living area, and then you have the back for the cows, for example, or for grain, and then you have a silo, and it's all kind of just built together, and you can kind of see how it grew. I hope that kind of answers it.
Yeah, I mean, I think for me also the rural, in some sense, it is even sort of an apparatus of maintenance for urban environments. So that's also an aspect. And I think in my work, I try to since I'm a craftsperson or carpenter, I'm also quite interested in these forms of knowledge and epistemologies that exist within the rural, even less and less today, as sort of open technologies that are, that go beyond sort of hierarchical knowledge regimes, who controls sort of the black boxes of knowledge. And I think therefore the rural can also help us to imagine sort of alternative pathways.
I mean, I would just add, because I think those are all great answers, that between the urban, the rural, and also just across different cultures, reaching kind of those very remote areas, that the cultivation of matter is about biology and is about architectural material, but I think it's also about knowledge, and finding ways to not just appropriate ideas and use them in different contexts, but also to find ways that knowledge is a form of exchange, and the fertilizing that we're doing as architects is contributing to societies, which, I think could take place in many, many different forms, but it's important that we hold that at the heart of the work that we do.
Yeah, we have another question there.
If you're creating conditions for life to exist in, and that's what you design, how do you decide what conditions are going to suit life to evolve? Because life will adapt to many different conditions, so we have to be selective about what life we want to live where and the kind of ecosystem that we're building. How have you thought about how we'd be selective about what conditions and ecosystems we want to build?
Yeah, so I'll just start with, I think that sometimes we fall under the trap, and I understand the start of it, but we fall into the trap that we need to control how life begins to exist. And there's really interesting elements of designing life within architecture which has repercussions which are not favorable. Things like mold inside homes, which is a real health epidemic. But I think what we need to pay attention to is how to design the conditions for life to still have agency. So oftentimes choosing, what kind of nutrients, the presence of water, all these things. But I'm not sure there's always, perhaps there is in a medical kind of application or in an agricultural application, a reason to tailor specific forms of life. But I think what we do as architects, just like we shouldn't control humans within their spaces, we just design a possibility, a spatial possibility, and then the humans have agency for that same kind of parallel to happen in an ecological sense, that we design the possibility for them to have their own agency and to take part in this multi-special existence.
I think for me it's also, I mean, you can attempt to create the conditions for a certain form of life to exist, but in the end, I mean, it's complicated, since like on the other side, like recursively, it can also trigger, ecological catastrophe, sort of, if you look at the rare earths extracted to provide sort of compounds for technological apparatuses. So I think for me, it's actually more about like experiments. It's a lot of tacit knowledge that you actually would have to experiment with to find that out, I think.
I mean, if I can jump at here, or to complete or follow up this question, because your last two, Paul and Luisa, you explicitly use the term uncertainty, right? And this is more or less in this condition. But we know from quite a few post-human theorists that advocates for uncertainty as a way to decentralize the human figure, but also warn us that we cannot just embrace uncertainty in a just positivistic ways, right? That this is every uncertain future, it's going to be better simply because we don't know what it's going to be. So in that question, I would like you to elaborate more on that sense, because as I explicitly you use this term, what can we understand as a kind of a sort of ethics of uncertainty, or if we should have a plan B or correction along the way if things turn out not as they should, then they are not uncertain anymore. So I don't know how you can handle this paradox.
I would say at the moment we're in a phase where there's a lot of tryouts happening, experimentation happening, and we need to accept failure. But of course, accepting failures also depends on what scale this happens. But if you start cultivating something and start being aware with the things and giving this, like in fermentation, you put everything, there is kind of a structure. You have the vessel, you put everything together, you put salt in it, and then there's more than an outcome. If we try to translate this into an architectural practice, of course, there's also some structure needed. You can't just say everything is in uncertainty. So I think you can set the boundaries by, I don't know, giving still certain infrastructures or possibilities, or people and humans, more than humans, being able to encounter. But I think it's, if you value the process, this is the possibility where it's more about the process itself and not about just the outcome. If we're just outcome driven, then uncertainty will maybe kill us. But yeah.
Yeah, I think very similar to that, where like, I don't necessarily think the outcome is the uncertainty, but like it's a process, and we're just uncertain about the process itself, and there are different temporalities. And also, like, to the question earlier on, like, what kind of life are we cultivating? I don't think we need to answer that, because if we design a system, if we design a process in which we can shift the outcome every now and then to fit what's best for the kind of multi-species inhabitants, whatever you want to call it, then we don't have to be like, oh, this is our outcome, but we can be like, okay, we're working step by step towards it, because, like, as you kind of said, we don't have really a plan B, right? Our cities are gonna just get worse and worse if we don't do something now. Similar with, like, what other people have already talked about as well, like, if the outcome should be no more capitalism, the outcome should be rethinking these structures of ownership and who owns what, because that's the thing that is kind of like ruining everything that we're trying to do, and kind of hindering all of our proposals. Same with, like, the rural and city kind of connection, where one gets more and more money and one gets drained of money. It's just, yeah, the uncertainty is just specifically at certain parts, and then you go from there. Yeah.
Maybe we take one more question. Well, if not, I asked that. And after these two intense days, one question that is keep on rolling in my mind is that, what do we do now as an architects, right? So to some extent, we don't believe anymore in this centralized model of the master architect. You very specifically, Luisa, present the architect as a fermenter. So we are used to a model in which the architect is a globalized control person that can build anything everywhere using anything, right? So do we need to break down this model into not then focusing our expertise in, I don't know, typologies or certain kind of materials, et cetera, but being specific and start specializing architects among regions or among processes or among, I don't know, how do you see now? Because many of the, especially again, the last two of you were even proposing or showing images of community-driven designs without any sort of centralized authorship. So what do we do now, us, as within those contexts? I don't know if you can very briefly elaborate, each of you, out of this question.
Okay, so maybe I think that what we have to do is to still try to make new approaches tangible and try to show them an application. And it's like, when I've been trained as an architect, there was also always the given task. There's this empty site, there's this empty plot, design something, then materials have been added, and then you maybe thought about how you could build it, and that's it. And I think by bringing materials and everything and being aware of the starting point changes your whole design process. I think this is part one. If you think about if we have to build how we build it. And then if you look at those contexts, and if we recognize that there are already people working on, people using the space, or being in inherited buildings, I think there are possibilities where we already can set intentions, try to build differently. So I think we have to show it, so also we can gain some interest for other peoples, or show them how it can work so they can relate to it. Because I have the feeling also with all of those approaches, it's sometimes very hard for even people who are in this theme of architecture to grasp it. And I think what we have to do is to make it kind of tangible now, so that we can show, okay, we can work with this and this material, it is possible. If we enable this community to build something, they can identify with it. And then this might spread the word, and then it can cultivate maybe a different way of building practice.
I think first I would say that it would come down to sort of form communities of practice for me, to sort of democratize the whole building process. meaning also to sort of rethink our whole relationship to building restrictions. And yeah, by forming these communities of practice, also question the sort of role of craft, again with the crafts, but to yeah, give them sort of back their authorship in buildings, since their role has been like diminished, in my opinion, more and more, due to sort of standardized building components. So these are things I think I would tackle. Yeah.
Yeah, I think there's obviously many dimensions to that question. I think one of them, which just has to be there though, is this idea of being local. Our materials are just circulating around the world, and we have the same answers for completely different conditions. Buildings in Helsinki and buildings in Dubai look almost identical, and it just makes no sense. So staying local means, yeah, using kind of materials that are local, using knowledge that is local, and then actually addressing these questions of climate and of ecology. And any future architecture which pretends to be sustainable has to have that locality at the heart of its kind of design process, I think.
Yeah, I mean, I don't have that much to say to that as well. Like, I feel like the architect has kind of positioned themselves at this point in society where they are kind of like the master of the built environment, and nothing can be done without them. But I think to kind of go back to what should we do, I think just have a like really long look in the mirror, think about your position, your entanglement in the kind of like locality of things, and who you're working with, how you're working with them. And just don't take yourself as seriously as you think you should be, because you're not. Like you're just building. Yeah.
Love it. So please an applause to the participants for this session. And with that, we just close up this session, and we jump directly in the closing words, right?
So good afternoon, everyone, and thank you for your patience. As we conclude these 2 intensively rich days of sharing, of challenging and imagining, I want to thank each of you for bringing your full curiosity to the event, your critical thinking, and your care to this topic. We are really grateful for the support of the Engevente and our rector Ulrike Kur, the head of the Institute of Architecture Sam Jacobs, and a special thanks to Petra Gruber, the head of the Building Construction Department. There is also, thank you for the applause. I want to extend also a big thanks to Claudia Rösli, Andreas Korner, Anja Jonkhans, Valentin Bossard, and Michaela Karpov, whose support has been really invaluable for helping organizing everything. A warm thank you as well for the technician, Maximilian and Thomas, who are behind the scene and who moderated everything for 2 days. And again, a thank you for all our moderators and the scientific committee who helped us over all these months to select the abstract and bring this full program of 2 days. So I also applaud them. And finally, thank you for the guest speaker and the audience for joining us in Vienna, and some of you traveled from very far, so we are really grateful for that. So this year we gathered under a special theme, especially for Petra, cultivating matter, and we questioned if the future was alive. And for us as a team, or at least for me, this topic really questioned the very boundary of architecture. And I think that this can really inspire us. And through the event, we heard, we discussed, we seen, and we also touched some prototype that shown that design is not only with material, but it's also with life itself. And we can see that architecture and buildings can be ecosystems. We can co-create with living organisms, and we can take them as a full collaborator in the whole design process. So all this innovative solution that we've seen, this speculation for the future, I think they were all pointing out to one big shift in how we conceive architecture. Rather than a static object, we need to think in dynamic and adaptive system, deeply embedded in flows of energy, of matter, and of time that are often overlooked in our practice. So this shift is technological, but is also ethical, I think, and is also maybe a shift of perspective that we have depicted over the last 2 days. So cultivating matter is a matter of care and of committing on long term to embrace the maintenance, the repair, and the adaptation. I think this is also reflecting on the definition of circularity, not as a finish line, but as an ongoing dialogue between the organism, the building, but also between the inert and alive matter. And sometimes this boundary can be also redefined, as we've seen in one of the session panel. And also connecting the past and the future of these materials. There was something that we've heard during the first keynote of Carole Collet, talking about the layer of rhythm of life and the pace of cultivating, that I think make me reflect a lot as well on the way we practice architecture nowadays. And another term that was evoked, cultivating rituals that also accompany this whole approach. So I believe that all the research that was presented here, the prototype and the speculative design, answer a bit of question that we posed if the future is alive. I think we've seen that, yes, I think there is the positive answer to that, but we don't know if we are already there yet. And we were now speculating with Petra if, at which scale we really apply this aliveness in architecture. But I think it's really emerging, and it can be optimistic and playful, as we said multiple times. So maybe what matters now is also what we carry forward in the coming days or months, maybe until next year until the next symposium, how we carry these principles that we've explored into our practices. So maybe by embedding care into our process. This is a notion that really surfaced again and again over the last 2 days. Also in our collaboration, so to really embrace more transdisciplinary approaches and bringing biologists, material scientific, architect, landscape designer, and maybe philosopher into our projects. And also in our teaching to think in terms of flows instead of fixed form, to educate our students who will shape the future of the architecture field. And to conclude, I would say that cultivating matter is also ultimately about cultivating relationship with the environment, but also with non-human agent and with time itself. If you were not lucky, or if you were unlucky to miss some of the presentation, I'm happy to say that it was all recorded, so I believe in a few weeks or months we will be able to publish it on our YouTube channel. If you want to rewatch some of the presentation or maybe reflect on it, you can also find a QR code to the book of abstract on the table there and on our website. And we are really happy to receive any feedback, comment, or suggestion that you may have by email. And now to conclude this 2-day program, I would like to invite you to visit the Root Arc exhibition that is located right outside on your left in the atrium, if you haven't had the opportunity already. Yesterday we had Clemens Preisinger presenting as part of session 2 on bio-inspiration, our project. So I won't elaborate too much, and we will be here to answer any question that you may have. But our work was fundamentally inspired by this nature intelligence and the efficiency of growth strategy in plant root systems. And we really aim to translate this principle into new architectural possibilities using digital design, experimental fabrication, and using our biolab that we could have for 1 year. So this exhibition is kind of concluding our work, but we hope that there will be a next chapter that may come along in 2026. So thank you again, everyone, for your attention. And now we can open the small reception with some drinks right outside the atrium. And our team will be here to answer any question you may have for the symposium or for the Root Arc project. Thank you so much.